फेसबुक मध्ये ना बॅकग्राऊंड नॉईज खूप येते.
सर अनम्यूट कोणी नाही आहे तसं.
Good morning, dear Prajwal.
Yeah, good morning, sir. How are you?
गुड मॉर्निंग प्रज्वल सर.
We will start in the next few minutes.
Sure, I am ready.
Anwar sir, mute yourself. Shall we start, Anwar sir?
Yes, yes, we should start.
Adikar sir,
Yes, yes, sir.
you can start.
Okay, sir.
Principal sir, respected principal sir, can we start the session? Adikar sir, just wait a minute.
Yes, sir. Yes, sir.
Good morning, respected principal, sir.
Yes, good morning, Dr. Kumar.
Sir, can we start now?
Yes, please start.
Okay, sir. Thank you very much, sir. Sir, please proceed. Vadekar, sir, please proceed.
Sir. Good morning and warm welcome to all on the second day of first international webinar series on artificial intelligence in drug discovery. Respected speakers, dear participants, today we have with us eminent speakers Dr. Prajwal Nanikar sir and Dr. Arjit Roy sir. May I now request from Mr. Pradeep Bhavne sir to kindly introduce our first speaker of the session, Dr. Prajwal Nanikar sir.
Thank you, Adikar sir. Am I audible?
Yes, sir.
Prajwal, sir,
Adikar sir.
Prajwal, sir has joined.
Prajwal sir, are you there in the meeting?
Yes, yes, I am here.
Okay, thank you.
Thank you, Adikar sir, for giving me this opportunity. It is a great privilege to introduce today's first speaker, Dr. Prajwal Nanikar sir, who is going to deliver a talk on topic transforming drug discovery with advanced computational modeling. As AI has become an integral part of drug discovery today, as we are using various applications of machine learning and artificial intelligence to meet the challenges of computational drug discovery, computational methods are effective approaches to reduce the number of safety issues by analyzing possible links between the chemical structure. We have a very right person here with us to share his thoughts on this topic of prime importance. Dr. Prajwal Nanikar received his master and PhD degree from National Institute of Pharmaceutical Education and Research, that is NIPER Mohali, in 2010 and 2015 respectively. He is a recipient of prestigious DAAG fellowship, that is Deutscher Akademischer Austauschdienst, a German academic exchange service, to perform his research at Heidelberg University. He has 4 years of postdoctoral research experience in using various types of molecular modeling techniques to understand biological process. His research interests are computer-aided drug design, quantum chemistry, and multi-scale molecular dynamics simulation. He has been associated with many eminent scientists and has experience in diverse range of research projects and protein, DNA as drug targets. He holds 36 publications to his credit. With this short introduction, I will call upon Dr. Prajwal Nanikar to please take over the session. Over to you, sir.
Yep. Thank you very much, Dr. Pradeep. So thanks for nice introduction. So let me share the screen. I hope I have a privilege to share screen. Is it?
No, it is not visible, sir, yet.
Yep, I'm just sharing it. Yep.
Yes, sir, it is
I
visible.
hope it is fine. Okay,
Yeah,
great.
sir.
Yes,
Beautiful,
sir, it is.
beautiful slide is visible, sir, now.
Sure. So, yeah. So here you go, and I hope I'm perfectly audible also.
Yes, sir.
Yes, sir, you
Okay.
are.
Yeah, great, great. Thank you very much. So again, thanks for giving me the opportunity to present here. And as I can see, it is the first conference, virtual conference on AI in drug discovery. So it's really nice that you are conducting this conference and you got many eminent speakers on board. And hopefully, I will attend the coming sessions other than mine, of course. So I'm really interested in looking forward for those sessions. So related to my task specifically, so as in the introduction, I'm working as a senior scientist with Schrodinger from past 3 years. And so here is my email ID, which is pretty simple, prajwal@schrodinger.com, and here is the number, and that too is kind of simple, 99000900 and 33. So the idea is, so today I'm going to give you a brief overview of artificial intelligence enabled tool which are present in the Schrodinger suite, and what is the idea behind it and what is methodology behind it. So maybe because of time constraints or anything, if few of questions are not answered, or if you want to know more about it, or at any given point, if you want to have a discussion on your project, specifically computer-aided drug designing projects, please feel free to reach out to me on this email or on the phone. So basically, that's the idea. And so this is what the brief overview which I am going to cover, and I will tweak the presentation around AI in drug discovery. But again, before going directly to AI, first things we need to understand little bit basics of it, and what are the various tools which one could use it efficiently with the help of AI algorithms, and how one could accelerate the drug discovery. That will be the complete workflow of the presentation. So let's start working from really basics. This is what very in general we know, as I expect most of us are pharmacy student. So I'm also a pharmacy student, and I'm really glad and happy to tell you that I am a junior of Principal Dr. Samir Goyal. So and Dr. Amit Gangwal. So I mean, we study from the same college from now. So this is what is really basics of drug designing. Is whenever we talk about drug designing, so we talk about a drug molecule which has some pharmacological activity. And when we say some pharmacological activity, so that pharmacological activity is governed through some receptor. This is what we know hypothetically from our B pharma or from our basic education. So when we say receptor, so simple thing, just like antihistaminic drug like chlorpheniramine. So that has a target of histamine. So that is a protein. So basically, every pharmacological action is governed through some receptor or through some signaling pathway, and this is what is our target. So you can see here the protein somehow looks like this. So this is what the surface view of protein, which is kind of globular, very uneven in shape, which is always present in a motion inside our body, and because of its motion only, it it is able to do some action or its role in biochemical pathway. And the idea of drug designing is to modulate the action of this protein. So for example, if this protein is moving, so if this protein is moving, it follows certain biochemical pathway through which it shows some action. But when drugs come into picture and that drug or medicine gets bind somewhere in the protein, specifically inside the binding pocket, it arrests the molecular mechanism which is related for the disease or which is causing disease. And simple example can be fever, like whenever you have high fever, it's a prostaglandin which gets synthesized more and more, and our aim is to reduce that prostaglandin synthesis. So we take paracetamol or aspirin, and that acts on the receptor and it reduces the production of prostaglandins. So this is what the idea is, and now let's go one step ahead and try to understand what is happening exactly where the substrate or inhibitor or cofactor get binds, and because of that it reduces, it arrests the protein function, and so the show pharmacological action, and it follows the lock and key model. So same like you have a lock certain binding pocket where key gets fit, and as key you rotate the key, it opens the lock or close the lock, and this is what we need to do in case of drug designing. But the thing is very much tricky when you can't see basically. So the idea is to design a key against particular lock. But what if you can't see inside the lock? Then it will be really tedious to make the key for key maker. But what if tomorrow we get a microscope through which you can able to see how the locks look like inside? So if it is so, then definitely the task becomes very much easy in such case. So this is what the idea is. If you are able to see what is how the key looks like inside, then you will be able to see the, particularly you will be able to see what are the levers present. Not only that, you will be able to design a perfect key for that lock, and that can be very much specific to this particular lock. So that's what the idea of having software, specifically molecular modeling software, just like Schrodinger. So the same thing applied to the biological receptor. So with the help of molecular microscope called molecular modeling software like Schrodinger, you can see at the atomic level details of the receptors as well as the drug molecule or the molecule which you design, the new chemical entity. And with the help of this microscope, you could not only see in the structure of inside at atomic level or at electrostatic level, but you can also see the structure of your key or the ligand molecule or the chemical at its properties, electronic properties like positive surface, negative surface, hydrogen bond acceptor group, hydrogen bond donor group. So with this idea of looking into the receptor, you could able to design a better molecule against that particular receptor. So that's what the overall idea of developing software, all the molecular modeling software, to enables you to see inside the structure. So basically, when we talk about molecular modeling, again, this is what is very much basics of molecular modeling. So it is nothing but, when we talk about molecular modeling, it's a field which was developed since a really long time back. I would say 1960s. So, and it includes basically it's a interdisciplinary field of all the disciplines, basic disciplines like chemistry, physics, mathematics, and biology. So you need to have all that knowledge. Not only that, but you also need to have knowledge of computer science, at least something basics, so that you can handle the computers because it's a software. So, and so with the help of all that knowledge, we try to build a model of a molecule. So if you see this particular molecule which is in front of you, and this molecule, if you could identify, it is nothing but the 3D structure of paracetamol. So each gray carbon sphere represents the carbon, red represents the oxygen, and blue represents nitrogen, and all white color spheres are hydrogen. So 2D structures look like in the two-dimensional, but this is what the 3D structure of paracetamol is. So basically, we need such model to study its property, to see like how it behaves inside the body, and we try to do all those things in computers, and it correlates very well with the actual biological thing. So to study that, we need a molecular model, and this is what the field emerges from. So it took like 60, 70 years to develop the field, and we are now at the stage of the peak of this particular field of molecular modeling. So if tomorrow you say like, can I simulate or can I study the actual biological phenomena which is happening in the laboratory or in the body, can I do the same thing in computers? So the every molecular modeling scientist would obviously say yes, you can model it because the field has grown that much. We did that much advances in the field. So let me show you one of the example of it. So this is what the videos of computer simulations of water in liquid phase and water in gaseous phase. So let's see the left hand side video. It is a simulation in computers done by the software at, let's say, 298 Kelvin, which is almost a room temperature. So, and you can see here the central is water molecule, red color oxygen and white color hydrogen, so H2O, and which is surrounded by a lot of other water molecules in the surrounding. So you can see all are other water molecules. So I have focused on only single water molecule here. So in this simulation, what you can see, these dotted yellow color lines are nothing but the hydrogen bonding interaction. So there are intermolecular hydrogen bonding interactions are occurring between one water molecule and surrounding water molecules. And you can see, if you see these dashed lines, you will see there are four number of hydrogen bonding interactions which are occurring at any given point. So in the time scale, so you see like this is all moving, means this is the same thing, water is placed in the beaker, and then what is happening actually in the beaker in water when water is present in liquid phase. So this is how it behaves, and it forms four hydrogen bonding interaction at room temperature. That makes lot of interaction of water molecule which keeps water in liquid phase. But what if in the computers itself, by using mathematical equation, you increase the temperature to 368 Kelvin, which is almost the boiling point of temperature water molecule. So you see, like when you start boiling the water gets converted into from gaseous phase, from liquid phase to gaseous phase. And this is what happens in gaseous phase. It expands because of extra energy you provide in form of heat. So because of that extra energy, the hydrogen bonds, the number of hydrogen bonds start declining. So now you can see at any given point here, there are no four hydrogen bonds. It's two or three at most. So because of those less number of hydrogen bonding interaction, water starts converted into gaseous phase and start expanding. So this is what the phenomena which you can observe it at atomic level with the help of computers. So basically the idea of showing it is, this is what the power of molecular modeling tools or the softwares nowadays. So you don't need to go to lab and check what is exactly happening inside the microscope. The water, you can simply do it in the computers. And same thing is applied to the drug receptor binding or drug designing. So from where it all started, so I would say like it has started really a long time back. There was a scientist, Martin Karplus. So in 1960s, like he developed the force field to define these molecules in terms of molecular model with the help of computers. And like from 1960s he started doing it, and it took almost next 30 years to grow that field, develop it step by step, and actually implement it into the industry for drug designing projects. And the first successful example was reported by Abbott Laboratories for HIV protease inhibitor. So this was the first example of structure based drug designing with the help of computers. So you can see like how long the journey is to develop the field, and now we are in 2020s, and we are almost at the very advanced level where we could accurately predict whatever is happening exactly inside the body or protein. So that what it took the journey to be. And nowadays if you see like it was started with HIV protease inhibitor in 1990s, but nowadays almost every drug molecule coming into the market or in the clinical trial goes through the phase of computer aided drug designing, and there are number of examples for that. Here is a list, but that list is really small. It is a very huge list nowadays. Almost every molecule, I would say, goes with the process. Every pharma company spends a lot of money on the research and development, and specifically on computer aided drug designing. Every pharma company has the software like Schrodinger suite, and they have a research group who is dedicated for doing these computer aided drug designing studies. So why so idea of spending more money in computer aided drug designing is because if you look into the overall drug discovery phases, It's a really huge process which could take like 12 to 13 years of time to complete, from identification of target to the drug into the market. That includes initial phase of drug discovery and research, and then that also includes clinical phases, preclinical phases, and that's also huge time one need to spend on. So you could see this blue color is mostly a time which company spends on doing drug discovery research, initial drug discovery research, I would say, before putting molecule into the clinical phases. And it would also take 5 to 6 years of time. And if you see in the timeline, like it's really huge. And even afterwards for clinical phase also, it requires 7 to 8 years of time, and that you cannot really compromise on, because that's all predefined protocols. We need to spend that much of time in drug discovery, the clinical phase. But as a scientist, what we can do? Can we reduce this complete time period? Can we work on some methodologies or some phases where we can reduce it? So yes, we can do it by reducing the initial phase of drug discovery and research, means starting from the identification of target to the putting molecule into the preclinical phase. Because once clinical phase starts, like you can't really have any control on the molecule. The structure of molecule you can't play further with, whatever the IND approval you got it on, and you can't basically play with the molecule. It's the known thing is, in clinical phases, it's a molecule which play with the body. Means whatever molecule behaves, we can, as a clinician or as a clinical research scientist, we can only observe those parameters and note it down and learn from it. But if somebody asks you to modify that molecule, we can't do that. But we can do that in the initial phase of drug discovery, which also takes 5 to 6 years of time. So here is the input coming from all the computer-aided drug designing scientists as well as all other discipline scientists. We can work more on this phase, and we can do as much as possible to reduce this time span, to accelerate this particular process. So money wise also, you could see like around 40% of money goes into initial phase of drug discovery, and around 60% of money goes into the clinical phases. So that money can also be reduced, time can also be reduced, and we will see in the coming slides how it can be reduced. So overall cost, as per this particular article, you can see like it's around 2.6 billion US dollar, which would convert into around 18,000 crores of rupees. So, here our role comes. So if you segregate that the initial phase of drug discovery, so there are various steps where, as a computer-aided drug design scientist, we can have our role. It starts from the use of bioinformatics tools for identification of the potent target, their pathways, designing new molecules and testing those molecules, further modifying those hit or lead molecules and get it into the really up to the good extent, with not only the biological activity but also a good pharmacokinetic parameters. And once you have gone through the process of computer-aided drug designing, you come up with the molecule as best as possible and get IND approval for that and go for preclinical studies on it. Of course, this CAD methodologies includes, with the help of other biological disciplines or with the help of experimentalists, we need to optimize. It's a to and fro process. All scientists come together and work on this process to reduce the time scale. And why it is so? Because whenever we talk about drug molecule, it's not only about the drug receptor binding which we need to improve, but at same point we need to work on various aspects of the that particular molecule, like its stability, its safety, its toxicity, its pharmacokinetic property. And there are hundreds of such properties which you all need to optimize whenever whenever we design a molecule and put it into preclinical phase. Before that, we need to optimize all the properties up to the satisfactory level. So that is why there is very well known saying that designing drug is harder than rocket science. So if you see, like, I don't need to explain this thing, but if you simply compare from past 50, 60 years in post-independence era, how many satellites, how many rockets India has launched. If you see the ISRO timeline, you will see there are many rockets are launched as well as satellites India has. But what about drug molecules? So if you say if you look into the molecules which India has developed so far, so I would say there is no new chemical entity which is developed, though there are few molecules developed by Zydus, CDRI, but those are again either a derivative of natural products or phytochemicals or the derivatives of existing molecules. But in terms of new chemical entity, there is absolutely no. So here comes the responsibility as a drug designing scientist, that we need to work hard to get the first Indian drug molecule. So Schrodinger is also working on the same aim along with other pharma companies. So for that, Schrodinger comes to rescue you. So basically, Schrodinger is a huge software suite which has different type of models for not only small molecule drug discovery, but you also have modules for biologics design like antigens, antibodies, vaccine development, and as well as even materials development. So when I say materials, so we talk about drug polymer interaction, designing new formulations, studying those formulations with the help of computers. And not only that, even the in ISRO, like whenever they do materials research to design a efficient satellite material or rocket material, they use the Schrodinger software. I was even surprised to know that ISRO, Boeing are our customers, so who use the material science suite for their materials research for designing of better materials, and also the discovery informatics, which talks about big data analysis in the drug discovery research. So basically, this software can be used by various departments you can think of in our field of life sciences, I would say, or biology or physics. So basically, all the departments can use it. Whoever wants to do research and whoever talks about atomic level detail of the things what he is studying, he or she is studying, they can use the software in their research. So it is a really wide array of usability. So maybe I would not focus on all those modules because our aim is to stick to AI in drug discovery. So I'll just quickly cover main thing is AI. So in case of small molecule drug discovery suite, there are many modules which are available. For example, understanding protein ligand interaction, basically how ligand or how drug molecule fits inside the protein. We can study it using with the help of molecular docking as a methodology. So you can also include the flexibility of receptor in the molecular modeling studies with the help of induced fit docking. One can also use the information of ligand structure, means the existing drug molecules, to find out the similar type of drug molecules with the help of ligand-based methods like pharmacophore, shape-based screening, or 3D QSAR methods. Not only that, you can also identify. So if this comes into the target identification, when you have very new protein with you, then can I identify where the binding pocket is, where the potential drug can fit inside it? We can do that analysis. We can also design a new molecules to skip the existing patent with the help of core hopping technology. So this is what very well known in the industry, because they every time they want to skip the patent of existing molecules to get the IP of new molecules. So core hopping is the methodology. You can analyze the all the energy parameters of whenever ligand gets bind to the protein, what energetics is involved in it, which atom of the molecule is contributing more towards that binding or more towards that energy. You can also generate the library of molecules with the help of enumeration tool. And when I say generate library of molecule, means you can do various substitution in single click and it will generate like lakhs and crores of molecule in just single click. So this is what the power we are nowadays. And if you go higher on higher level of studies in molecular modeling, so it's a molecular dynamic simulations which we talk about, the real time scale simulation. So molecular dynamics is the one which I showed you for the water simulation in liquid phase and in gaseous phase. The same thing can be studied for protein ligand interaction, and you can see how in real time protein and ligand is binding together, how it is behaving. So it is the same thing if you do the experiments in chemistry or biotechnology lab. The same thing you can do it with the help of computers. It will save a lot of time, efforts, as well as money. So other than that, even covalent binding can be studied. For example, recently there was a structure which was reported for COVID-19 protein, and it was a covalent bound form. So basically, this is what you can also study with the help of computers. You can design covalent binders. This is also nowadays in very much in discussion. So in this way, there are various technologies which one could use with the help of Schrodinger suit. So I would simply stick to the molecular docking because this is the simplest form of molecular modeling studies which most of people use, where you have a structure of protein which is comprises of very uneven surface. You can see the 3D structure of protein here, and with very uneven surface, it has particular binding pocket, and in that binding pocket, we dock like lakhs of molecule inside it, and after performing molecular docking, you get the binding pose of all those molecules. And based on molecular docking score and molecular docking interaction, we finalize few hit, let's say this molecule, and then we try to perform various substitutions on this heat molecule to improve its binding, to improve its interaction with the surrounding amino acid residue inside the binding pocket. And in this way, after rounds and rounds of modifications and substitutions, we come up with the final molecule, which we call it as a lead molecule, and this molecule we propose it for the further pharmacokinetic evaluations. So this is what the technology of molecular docking is. And now you will say, okay, what to do with this in terms of AI? Like again, I would say we are slowly building it towards AI. So please be with me. So these are the technologies which we are going to use it in future in coming slides. So let's see. So when we talk about molecular docking, so it's a simple methodology to identify how a drug molecule or how a chemical ligand fits inside the protein structure to study its interaction. So it has different levels based on speed and accuracy of docking. So there are three algorithms which are present with Schrodinger, like high throughput virtual screening algorithm. It takes around 1 to 2 seconds for each molecule to dock inside the protein. Then there is a standard precision algorithm which takes around 20 seconds to 30 seconds for each molecule to dock inside the protein structure. And there is extra precision which has highest accuracy, but it would take like 2 to 3 minutes for each molecule to dock inside it. So you would say like why I am explaining this thing. If you are new to this molecular modeling field, or even if you are experienced in molecular modeling field, the time which I am focusing on is really considerable, because with the help of any other software, if you talk about, it would take, even for the simple molecular docking, it takes around 5 to 10 minutes of time for each molecule to dock. And here we are talking about fastest algorithm which could take 1 to 2 seconds for each molecule to dock. And why it is important? Because in drug discovery, we do screen lakhs, crores, I would say billions of compounds, like 100 billion compounds we need to screen. Then only you get one potent molecule which you could put it into clinical trials. So we need to study a lot of molecules, and with the help of existing technology, we should have that efficient algorithm. We just take like few seconds to dock at least this molecule. So that is the reason why we need efficient software like Schrodinger to study it. And coming towards the result tomorrow, I can say like, okay, it is faster enough, but how much about the reliability of result? So for that, somebody has published study in 2012. They compare various softwares like Schrodinger's Glide, then very well-known Gold software, and Dock and Autodock software. So out of comparison of all those software, you can see like the enrichment calculations given by this Glide software, orange color, is very high as compared to other software. So which says the reliability of result, that it is able to reproduce the exact crystal structure pose whatever you observe in experiments. So this is what I talk about the normal molecular docking, which is most of the people use this kind of general molecular docking like high throughput virtual screening, standard precision, extra precision algorithm. But when we talk about docking, so it's not a general docking of protein and organic compound, but there can be a instance when you are working on peptides or peptidomimetics, and in that case you need different molecular docking algorithm to deal with. So for that, we have a module called peptide docking. There can be instance when you start working on natural product or macrocyclic compounds. These are kind of big compounds with lot of restraints in the structure, and that needs a different algorithm to work with. So for that, we have different options. You may work on the covalent binders, for example, COVID protein or EGFR proteins. There are many covalent binders, so that needs a different algorithm to treat. So for every different type of molecule, we have different tools which are available. So this is what is necessary. So even for organometallic compound which has a metal in it like cisplatin, so such compounds needs a different treatment while doing molecular docking. So the overall idea is different type of ligands, because of their different chemistry, because of their different properties, they need different treatment. So tomorrow if somebody says, okay, I have one software which is able to do the docking of all type of molecules, then I would say that's not really good thing to deal with. You must have different type of algorithms to deal with different type of ligand, because every ligand is different in terms of chemistry and its properties. So this is what Schrodinger, that makes Schrodinger very much unique in nature. So this is what I was talking about, the methodology called docking. So just at this point, take home message is Schrodinger has a docking protocols, various docking protocols to deal with it. These are tremendously faster, can dock molecule in a second, 1 second or 2 seconds, and it is very much accurate. So these are the take home points you just remember at this point, and then I will tell you how one can use this. Then second thing comes into picture, which is very well known in terms of molecular modeling studies, is QSAR, quantitative structure activity relationship studies. This is what very well known in pharmaceutical chemistry or in the computer aided drug designing community. So whenever we talk about QSAR modeling, it's about the prediction of biological activity of a molecule by using various molecular descriptors which one could generate with the help of molecular structure and the software. So for example, tomorrow, and here we learn it from the existing data set. So you have an existing data set of, let's say, 10 compounds, and you know the log p value for all those 10 compounds. And then what you do, you see those all 10 molecules, look for the common features, common substitutions, common structures which are present in this molecule, and see its log p value. So you can basically make a relation out of it, how log p value is changing as you change the substitution. And this thing you learn from the existing data set of the molecules, and then you design something new molecule out of that learning, and then you try to predict what will be the log p value of that particular molecule. We do in the past, scientists used to do with the help of paper and pen, but eventually they have developed a software for doing all those things, and this is what the QSAR methodology is about. So as I told you the example of log p values, similarly you can also predict the biological activity or IC50 value of those compound with respect to the position of functional groups or various type of functional groups which are changing. We call it as structure activity relationship analysis, SAR analysis. So you can predict various type of biological activities with the help of existing data. You use QSAR technology and predict the activity of new molecules. And how accurate it is, that is a matter of this particular methodology as well as the effort of scientists, the input or the data set which the scientist has. So that's what we are is the nutshell and crux of QSAR activities. So we predict a biological activity of the molecule with the help of existing data. We try to fit this curve to the linear line, and it is simple as that of straight line equation, which is y is equal to mx plus c. So where you have x is the dependent variable and x is independent variable and y is the dependent variable for this straight line. So m is the constant, c is the constant, and as x changes, which is an independent variable, y also changes, and y is your biological x. So it's a simple form of QSAR equations. So basically what we can use it in a QSAR equation is, you have the structure of molecule with you, 2D structure of molecule, it can be 3D structure of molecule. You know what are the biological properties of those molecules like IC50 value, log P, its solubility, stability, or many parameters that you know. You calculate then all the descriptors like what is the volume, what is the area, what functional group it has, how many functional group it has. In this way, there are thousands of such properties are available We calculate those properties with the help of software, and we try to use various methods for QSAR modeling like multiple linear regression, which is MLR, PLS, partial least square, principal component analysis. In this way, there are many such methods which were available for doing QSAR modeling. And in this way, finally, we predict the biological activity of new molecules. So what is the workflow which we follow in terms of QSAR modeling is it is in general, and again you will be surprised to know that we are going to use the same methodology later on in terms of AI applications. So initially you get the structure of molecules, basically nothing but the data set of molecules, let's say 10 or 50 compounds you get. You try to look for the biological activity of that compound. There are certain parameters which we always need to look for, like it should be from the same assay method, preferably from the same lab, it should be reported in same paper. Then with the help of computers, we do generate descriptor for the data set of molecules, and descriptors means molecular weight. It's how many number of functional group, how many single bonds, how many rings. In this way, there are many such molecular descriptors available. I would say thousands of. We generate those thousands of descriptors, then we define training set and test set. So training set means out of 50 molecules, let's say 30 molecules, you put it as a training set. Training means software learn from those molecules which are present in the training set, and it generates a QSAR model. So QSAR model means nothing but an equation in the form of y is equal to mx plus c, and then it tests that model for the prediction of activity on the test set molecule. And we do analysis of this model, we validate it in all respect, and then we once the model get validated. So here whatever model I am saying, it is the mathematical equation. So we use that equation for prediction of the biological activity of new molecules which you are going to design. So here comes the crux. So scientists use our brain basically to define this test set and training set molecule. So this data set is constant, descriptors are constant generated by the software. So each molecule has a set of descriptors, and then those are constant. But the test set and training set is the main important thing which scientists need to decide, and scientists need to define these training set molecules and test set molecule. That's very, very tricky. You need a lot of experience of doing that. And generation of model is again the matter of computers and mathematical equations. So here comes our role. So everybody has their own way to basically design test set and training set molecules. For example, a layman or astrologer can define its own way for designing training set and test set molecule. So astrologer learn various features from the face or face recognition techniques with the help of training set molecules, and then test it on the test set molecule, and then apply this. It generates a QSAR model. It is nothing but the QSAR modeling, and apply it on the new data set to predict your future based on your facial lines or based on your palm lines. So this is how it works. But in terms of scientists or in the chemoinformatics or in computer aided drug designing, it's not human, but it's a molecules, small molecule structures. So based on those structure, we need to decide which are the training set, which are the test set. We need to learn from training set their biological properties, their structures, and we need to apply that model on test set. And then finally, once we can predict the biological activity of new molecule. So that's what the approach of scientist is. And here comes the descriptors. So descriptors are nothing but the properties which are for the structure of molecule, which we can calculate, as I say, molecular weight or number of carbon atoms or number of hydrogen atoms. So all those properties we can calculate. So initial days, like there were 1D properties which are generated from the SMILES format, single string of the molecule. Then there comes 2D properties which are generated from the 2D structure of molecules. Then one step ahead, there are 3D properties, there are 4D properties, which one could calculate nowadays. So you can see, like, over the period of time with the advancement, we could compute more and more number of properties. You can simply correlate with your experience with watching movies. So in past days, you would watch movies in 2D, Then eventually it turned up into 3D. So you have a goggle to see the movie and you get that 3D effect on it. But that's not the end. Even you have some 3D theaters or 5D theaters where you also feel whatever is going on. Like you have a shaking chairs, you have a sparkling water on your face, which gives you more and more experience. So this is what the same thing is applied in terms of descriptor. So day by day, it is increasing. The overall matter of telling all this story is we started from something which was very less, but over the period of time we started growing, and with the growing things we need really various methods to proceed further with. So what are those methods basically? So you see, like in general QSAR modeling, if you have less number of molecules like 20 to 100 molecules, you could use the existing traditional QSAR method with traditional mathematical analysis or statistical analytic techniques like PLS, PCA or MLR, these kind of techniques which you could use for less number of molecules, for less number of descriptors. But over the period of time, you could have larger data set like more than 100 to 1000 molecules. In that case, you need machine learning models to train your data set because it's tricky to train the data set. So you need various advanced models. There can be a situation nowadays when you have like 10,000 lakhs and crores of molecules and their data is available. In such case, you also need again efficient methods like deep learning or deep neural network or artificial neural networking or convolutional neural network. You need various type of advanced method to train the data set to learn from it, because with the help of simple visualizing or using traditional techniques we can't do it. So this is the point where it emerges the use of such a methods like machine learning, deep learning or artificial neural network to implement in the field of drug discovery, to understand this data, to learn from those data. So for that learning, we have a module called AutoQSAR. So just now I told you there is a protocol which one need to follow to build the QSAR models. And here I am coming up with the tool which is called as AutoQSAR. And here when I say auto, it means automatic. So what is automatic basically? So again, it's the same QSAR workflow. You have a data set, and now I am talking about data sets of thousands of molecules. So this is, though it is a simple QSAR, but now it is for the extended data set. You generate descriptors with the help of computer, but now when it comes to the thousands of molecule, as a scientist, like you can't simply assign test set and training set for thousands of molecules by visual inspection. You need efficient tool, and this efficient tool is called as AutoQSAR. So basically what happens here, which I was emphasizing on, so AutoQSAR is the automatic technique which does test set and training set with the help of all possible combination and permutation between the molecules. So so idea is, in normal QSAR techniques, you follow this workflow, you generate the best model out of it, you generate various QSAR models using various test set and training set for small data set. And you do it multiple times. Sometimes you do it like 10 times, sometimes you do it hundreds times, sometimes you need to do it thousands of times. So here number of times, number of iteration matters. So as a human, you can click 1000 times button. It takes lot of time, your effort, you need to shuffle the data set every time. So I remember in my days, like I would require a 6 months of time period to develop one best QSAR model out of it, out of the existing data set, and that too like a small data set of less than 100 molecules. So that was the time. But nowadays we have huge data set. So we need we use advanced techniques, and here comes the machine learning. So with the help of machine learning, with the help of software, we automate this process. So now you just need to submit your molecule to the module called AutoQSAR, and software takes care of this part of generating the descriptors, defining various test set and training set, generating models, checking that model whether it is good or not, and again go back, again shuffle the data set, again generate new training set, new test set. And software does it for thousands of time or lakhs of time until and unless software gets the best model out of it. And finally, when software gets best model out of it, automatically it gives you the output of those best models. So that you can take. So means the human work is getting reduced here because software is doing all that iterative thing and gives you the final best model as a output. So here this is called as automatic QSAR module, which does the thing for you. It gives you output of various best models and all the statistics related to that, the best fit line. So whatever is best, you will get it as an output without doing all that human manual repetitive work. It also gives you the pictorial representation of this QSAR model to tell you that which groups or which atoms are there for the increasing activity, which atoms are there for decreasing activity. So you could see here blue color is for increasing activity of molecule. If you do further substitutions here, then definitely the activity will increase. So you get that pictorial view also, and this is very much unique to my knowledge with the Schrodinger, that makes the life of scientist very easy in terms of QSAR modeling. So now comes to the artificial intelligence. So this is what the existing knowledge we have. So if you again go back to the first learning which I told you, take home message was there was a docking algorithm which is efficient, accurate and faster enough. Then there comes the QSAR method, which is there, and we have auto QSAR tool which does all iterative things, repetitive things by its own. You don't need to do that thing. You just need to submit your molecule and software give you the best results. So how I can integrate both techniques with the help of AI to use for the huge databases. And when I say here huge database, I talk about 100 billion molecules. So 1 million is 10 lakhs, and then thousand it's 1,000 times is 1 billion, and I'm talking about 100 billion. So just think of this number, and we need efficient techniques for this. And why do we need efficient techniques for this? So the answer is simple. Day by day, the number of molecules which are present in the database, which are reported, people are synthesizing it, people are designing it, is increasing tremendously. So so this is what the chemical space which is estimated is 10 to the power 60. So now you can imagine how much there are 60 zeros on 10. So what could be the number? It's not even billions or trillion. It's way beyond that. So this is the chemical space which will be soon, and then we need to deal with that chemical space if you want to find out that best drug molecule out of it. So to achieve that goal, to screen as many as possible, we have different methods which are even faster, like molecular docking, pharmacophore, shape-based screening, use of GPU computers. Nowadays, GPU is in a very much discussion because it is very faster than CPU, normal computer. So if you see in tentative timing, like it would take around 10 seconds for each molecule to dock. And if you talk about docking many molecules, you would see like how much, just here is the simple statistic of 500 million. 500 million is just half a billion. If you want to screen half a billion molecule on single CPU, it would take 158 years of time just to perform docking. And like docking is just a tiny step of drug discovery. As such, drug discovery takes 13 years of time. And here, just to do docking, if somebody asks you to spend that much time, it is not really worth. And so in this way, like the idea is like even the fastest algorithm, fastest techniques, whatever we have nowadays would take years and years of time to screen simply half a billion molecules. And I am talking about screening 100 billion molecules. Then only the things will be achieved. So what does it say? It says like there are methodologies available, but because our chemical space is increasing so much nowadays, so we need efficient algorithm. Otherwise, if you simply stick to these traditional methods, it will take hell lot of time. And we don't want to spend that much time because the COVID scenario, how bad it was. If you could wait for 15 years of time, probably the human race was gone. But with the help of efficient computational techniques, we would be able to even generate a vaccine, design a vaccine within a span of 8 months. So this is how we need to accelerate, we need to work on it. So because the buzzword is AI, so let's try to look into that buzzword. So there were many startup companies which started working on AI, and I'm talking about, so there are in many AI even applications that are there. Even our simple Google Map is a AI application which predicts the traffic and all, and time required to travel from one point to another point based on existing traffic. So in this way, they have also implemented the AI algorithms in drug discovery for designing good molecules. And even there was a first, this was the news coming from Extensia, and they developed the first drug molecule based on AI, and they put it into the clinical trial. That was really buzz that time. Schrodinger also had developed various AI techniques in drug discovery. They have collaborated with many pharma companies like AstraZeneca and US universities to develop that platform and use it for the drug discovery. So in this way, there are many companies which started working on. So I would not comment on whether how efficiently it works, how whether it is just a buzz or whether it is just a hype. But we need that efficient algorithm, and we need to implement it in drug discovery. That's my motto. So Schrodinger come up with their own AI algorithm by using the existing techniques like docking and Qsar with the help of these two models. So how it works basically? So basically here I am talking about the data set of 100 billions, huge data set. So basically what software does, you take, you give that data set of 100 billion molecule as an input. Okay, so if 100 billion is difficult to imagine, I would reduce that data set to 1 lakh molecule. 1 lakh molecule. So let's say you give 1 lakh molecule as an input. So just focus on the number or percentage, and then you will understand what is the scalability of this method. So you give 1 lakh molecule as an input. That's the only your task, just to give the input molecule. That's it. Once you give that input molecules to the computer and software, what software does? Software randomly picks around, let's say, 1,000 molecules out of 1 lakh molecule. Just 1% of molecule, let's say 1,000 molecules, software picks randomly. Okay, pick it, and software dock all those molecules, like 1,000 molecules. Docking of 1,000 molecule, just 1 second for each molecule would not take much long time, just 1 to 2 seconds. Like it would take 1,000 seconds, or at most 10,000 seconds, and that's not that just few hours or a few minutes. So software dock those molecules inside the protein. Software gets the docking score of all those 1,000 molecules. Software goes to then auto QSAR module. So here it use the docking module, get the pose, get the docking score, then submit it for auto QSAR calculation, for generate various descriptors automatically, and predict, generate the QSAR model for predicting docking score of the existing 1,000 molecules. It iterates this process thousands of times to get the best model out of it with the help of auto QSAR tool. Use the best model as a output and see if that model is good enough. If it is good enough, software algorithm checks that thing based on certain parameters. If that model is good enough, then it will proceed further for hit identification. If that model is not good enough, then software goes back. So for going back, it needs that artificial intelligence based on some parameters. It will check whether that model is good. If it is not good, then software will again go back, pick another set of 1,000 molecules from the data set, existing data set, run docking, generate thousands of QSAR model automatically, pick best out of it, check if that best is really good or not. If it is good, proceed further. If it is not good, then go back and do the same thing again. So here the idea is, with the help of AI algorithm, this software does this thing number of times, and after doing this number of time automatically with the help of its AI algorithm, it gives you the best model out of it for prediction of docking score, and then software use that best model for prediction of docking score of 1 lakh molecules which you have initially submitted, and see like whether you will be able to predict the docking score for 1 lakh molecules. So ideally you don't need to docking for 1 lakh molecules, you just need to do docking for 1,000 molecules. And the docking score for 1 lakh molecule will be predicted with the help of QSAR technique and identify the good molecules out of that 1 lakh molecule. So now just scale this number to 100 billion. Out of 100 billion, there will be around 1 lakh molecules which will be docked. Those 1 lakh molecules will be used for training purpose, iterative process, hundreds of time, and then give you the best model out of it. And then you predict a docking score of 100 billion molecule using this technique. So this is what the AI algorithm which Schrodinger has. It is working very efficiently, and if you see how efficiently it is working, how much acceleration it gives, so just simple by doing docking, if you want to dock 1 billion molecule, it requires 3000 days, and goes on, if you want to screen 100 billion compounds, it would require 2,90,000 days of computational time. But as AI come into picture with the help of active learning, right, this 2,90,000 days reduced to just 74 days. And you can see 1 billion compound can be screened just within 5 days of time period. So this is how it accelerates. So if you use simply stick to the docking and you want to screen really huge data set of 100 billion compound, it would take ages. I would say like it just a 2,90,000 days. You just calculate how many years it is. And now we are talking about efficiently just get reduced to 74 days, and for 1 billion compound it's just 5 days with the help of existing infrastructure. So that's what the idea of accelerating the discovery process. Here is one of the simple statistics for screening around 30 lakhs compounds using this technique. So why they took this 30 lakhs compound? Because 30 lakh is a reasonable number for any academic research institute to work with. Any faculty or any researcher can work with that many computers. So you just need one GPU computer and 30 CPUs. So nowadays this is also very common. You can find it in any national institute. That's not very costly. It would take like 5 days of time to screen all the 30 lakhs compound with the help of this algorithm. So this is what is the technique which we are talking about, which will accelerate the process of drug discovery and screen huge data set for you. So in this way, like Schrodinger has plethora of tools which helps you to basically deals with all type of problems which are coming up in the computer aided drug designing. You can give your all the input for all the understanding or studying all the properties of molecule with the help of various tools. So that's the plethora of tools. I have shown here a few, but there are more than 100 of such tools. So so main thing comes into picture is you can say, okay, you are talking a lot about various tools, their usability, but whether it is really useful or not. So for that, here is the example in which it is a kind of success story in which Schrodinger itself worked on this particular project along with collaboration with Nimbus Therapeutics. So in this particular collaboration, Schrodinger was a computer-aided drug designing partner and Nimbus was a experimental partner. They work together on the target called acetyl coenzyme A carboxylase, which is ACC. So ACC, acetyl coenzyme A carboxylase, is very well known in liver diseases, specifically NASH, non-alcoholic steatohepatitis syndrome, NASH disease. And we have often heard the ACC enzyme in our biochemistry second year B Pharma. That it is involved in lipid fatty acid synthesis and fatty acid metabolism. So it was a potential target for every pharma industry to work on. So Schrodinger also came into picture and they work with Nimbus Therapeutics. Initially, they generated around 1.3 millions of compound virtually with the help of computers. With the help of enumeration tool, they screen with the help of all the computational techniques. They got the heat molecule with let's say 8 micromolar of activity. They took this heat molecule further, optimized that heat molecule, got the activity of lead molecule up to nanomolar range, let's say 200 nanomolar range. They took this lead candidate and further optimized that lead molecule to improve its binding as well as pharmacokinetics. And they got the final molecule with less than 10 nanomolar range. So they got this molecule, they filed for IND approval. They got the IND approval also, and they afterwards, because it's a software company, so they could not do clinical trials. So they sold this particular molecule to the big pharma company for huge amount of money. So the point here to be noted that not the protocol. So protocol of course includes use of advanced computational technique to accelerate the process with the help of all the biotechnology experiments or all the experimental techniques. But here I would like to point out the timeline. So you can see this is the timeline shown here in month. So within a time period of 12 months, they got something from really scratch, initial idea up to the IND approval. And if you remember our drug discovery pathway, which I showed earlier, we talk about initial phase of drug discovery up to preclinical phase require 5 to 6 years of time. And here it is just a 1 year of time. So this is what we call it as acceleration of drug discovery with the help of advanced computational tools along with all experimental study. So this is what the acceleration we would require nowadays to do the drug discovery faster. It will not only save our time, but time is money, so it will save our money also as well as effort. So in this way, so the molecule which I was showing is basically a molecule given by so here you go. That particular molecule is this one, Nimbus Therapeutics and Metabolic Disease. So currently this molecule is in phase 2 clinical trial. So here you can say various stages like initial drug discovery, preclinical and clinical phases. So along with that molecule, Schrodinger collaborated with lot of big pharma giants. List you can find it here. And there are many molecules which are in initial phase of drug discovery or advanced phase of drug discovery, even in the clinical trials. So the currently that molecule is in phase 2 clinical trial, but other than that, with Agios Pharma, we got 2 molecules which are FDA approved. So soon those molecules will be in market. So this is how Schrodinger is not only working on the software development, but also in the implementation of that software along with pharma companies to accelerate their research. And here I would like to specifically mention about this particular company, which is Spark. So if you know Spark, if you have heard of it, is a R&D subsidiary of Sun Pharma. So the name itself is Sun Pharma Advanced Research Center, Spark. So we have collaborated with Spark also, and so this particular molecule is on neurodegenerative diseases and in initial phase of drug discovery. So we hope that if this project gets successful at the end, then this will be the first Indian new chemical entity which will be in the market. So in this way, there are many molecules which are in the various phases of drug discovery. And one of the recent, according to recent news, like there is a target MALT1 in which we are almost enabling the IND studies for this molecule where Schrodinger, this is the Schrodinger own molecule. So afterwards, like Schrodinger started their not only working on the software, but we also started our own biological unit to test the molecules. We can perform all type of experimental studies on molecules. So this is our own molecule which way if get successful in clinical trials, then it will be the molecule from Schrodinger. So in this way, Schrodinger has many big multi-billion deals with many companies like BMS, Bristol Myers Squibb, and AstraZeneca, Bayer, and also Schrodinger participated in the COVID alliance of big pharma giants to provide them all molecular modeling supports. And this is what is the philanthropic initiative, and no company will get basically the IP on this drug molecule which they have discovered on COVID-19 because it's a philanthropic initiative. With this, I think I just stop the presentation. I guess I am on time. And finally, I would like to acknowledge my team who are working here in Bangalore office. So we all are located in Bangalore office. We are a team of 9 people here. Out of that, 5 are scientists, and we are all there to help you out in terms of any project related queries or any scientific discussion. If you have, you can reach out to us. We are always there to help you out. Additionally, before concluding the session, I would like to mention that if you find the software interesting, if you want to use it for your research purpose in academics, so we do provide 1 month of free evaluation of license. So that free evaluation means whatever modules which I have discussed and other hundreds of such modules, that all modules comes for free for 1 month on your specific computer. You can use it, you can do your project, you can publish your research if possible. We are happy to have it. And we also provide the free training to the user who wants to learn it. During that period, we provide all the scientific support to the person. But the only thing here is, of course, access software is very costly if you want to buy it at institutional level. But for 1 month, we give it for free for learning purpose. So only request from our side is to make as much as possible use of this free time. So whichever things comes to free, like we don't respect or we don't value that thing, but this is what our request is. Please value that free evaluation if you are availing it, because everything comes to you for free for 1 month. So use it as much as possible, learn as much as possible, and get your research published. So in case of if you want it, like just contact me on email ID prajwal@shorigar. And finally, with this, I would say thank you to all of you and for sincerely listening the presentation. And again, I am thankful to the organizers of this event, nice way and communications all. And if you have any questions, I am happy to answer it. So thank you very much again, all of you. Please be in touch.
Yeah. Thank you, Dr. Prajwal, for very beautifully explaining the content through your lucid slides and infographics. It was very down to earth type of a step-by-step explanation you have given for that. Now I hand over to Bhavna sir.
Thank you, sir. It was a really treat to watch presentation. There are questions from the audience. I have unmuted Ajay Kumar Gandhi, sir. Please unmute yourself and ask your question.
Yes, sir. Thank you. Prajwal sir, will you please go to the Qsar flow slide so I can better answer a better question them on that basis?
Okay, sure. Let me go to QSAR slide.
I wanted to know what different kinds of regression techniques that are provided there, and what algorithm also that I wanted to know in the proprietary software that you have provided. And the third question, that what kind of graph in graphical format we are going to get results, like for William plots, which type of graph we are going to have to know outliers and those things.
Okay, so I mean, it's really good to see that question because I guess you belongs to that community of QSAR modeling. You look for various statistical techniques,
Yes.
and then my answer is all. So you can do all type of statistical techniques in analysis of QSAR or generation of QSAR model like MLR, PLS. Not only that, even deep or deep learning
Neural
or machine learning techniques or neural networks also you can use. You can generate numbers of descriptor that only not limited to chemoinformatic descriptor, but you can also generate quantum mechanical descriptor, molecular mechanics, fingerprint based descriptors. So maybe I am missing some of the names specifically, but I would say whatever you have heard so far in the QSAR technique, all methods, all type of descriptors are all present here in this tool.
No, actually, when you were explaining, no, you just stated like y equals to mx plus c. That's the linear mode of regression. So I thought that just a linear mode of regression was given. But in the next slides, you explained that you can have the access to, like you can choose a deep neural network also, random forest, and these things also, support vector also. So accordingly, we are going to get results in the software. And what kind of then graph we will be provided by the software for the Williams plot and other type of graphs?
So basically, software gives you the data in the format of table. So you can see first thing is like you get different models. Okay, here. So you get different models, their names, which type of descriptors it has used, what is score, what is Q square and R square. That's very generic thing.
Yes.
In one layer of detail, you get the all training set, test set data, their predictions, their inputs and errors, statistical parameters you get, okay, default. one step for the analysis, you get this particular graph, correlation graph for the activity and the predictions. Other than that, means you get all that data in terms of text file or Excel sheet, and if you want to do further analysis, you can do that. But by default, software gives you these three data. Other than that, like this is what is very important observation which software gives you, the pictorial representation of QSAR model, which functional group is important, which is not important for the activity. And I think this is very unique. Means, to my
Yes, sir,
knowledge,
yes.
like I haven't seen such a pictorial representation in any of the software which I have used. So that makes
Yes,
it unique.
this is the only purpose of QSR, that you have idea of positive and negative things which work for or against the activity. And sir, regarding to this only, is this software integrated with scaffold hooping or something like that? So we can predict some further future molecules which will be best suited based on this data set which we have used, or the scaffolds which we worked with.
Yes.
Is it integrated with?
Yes. So as I said, maybe these things did not. Sorry. Yeah. So basically, yeah, now it started working. So you can see like it has all the modules that also includes scaffold hopping, means score hopping, shape-based
Okay.
screenings, pharmacophore-based screening. So simple answer to your question or even future question. So if you ask me, can I do this thing with the help of Schrodinger suit? And this means anything, okay?
Yes.
Whatever you put there, and my answer will be yes. So I say there are more than hundreds of tools which are available to enable the drug discovery, drug designing process. So everything is there. Maybe I cannot show it here, but everything is there.
So, Dr. Prajwal, I would like to add one point here with your kind permission. For his, what he's asking that type of graphical representation. So I think Mr. Gandhi, you can go for I think Matplotlib and Seaborn type of functionality, which is in GitHub or other type of AI tools. Through that you can have a lot of different visuals of data outliers and pens and all those things. So two concepts are there, that is Seaborn and Matplotlib, if I am not wrong. Dr. Prajwal, am I right? I think.
Yes, exactly. Means once you get the data in your hand in terms of text or Excel, there are like plethora of software tools, programming languages are available. You can use it. Like it's not really limited. It's all thing one can use. You just need data.
No, sir. My question was because just I wanted to know what features Schrodinger can provide to me. So when we wanted to purchase it, so we must have some rationalization to provide to institute. So another question related to this regarding purchase: is it made available on GEM, government e-marketplace?
Yes, it is
Yes.
available. So you just need to communicate with us that how you want to proceed.
Okay, thank you. And sir, just one question. In one technique, you've shown that you are going to use docking score as instead of bioactivity. Am I right?
Yes.
Whether I understood that correctly, that instead of bioactivity, we can use docking score as the thing that we can use as the characteristic of the molecule?
Yes, right.
Yes. Okay. Thank you so much. Thank you.
Sir, one question is from my side, if you permit.
Yeah, of course, please.
As the leading pharmaceutical companies are using this computational tool today for discovering the drug, and even after launching the drug, after doing so much of the study, the drug has been market recall. Okay, so whether we are not up to the mark in predicting the whatever the consequences which may happen after the administration of the drug, what can be done with this respect by using a computational method?
Yeah, I think as I said, like you can predict as much as possible. Not only predict, but predict accurately as much as possible. But again, when it comes to the clinical phase, it's literally out of our control. So afterwards, once we administer drug, it's the drug which plays like a monkey, I would say, some crazy monkey. Drug can go here, there, produce pharmacological, produce toxicological effect, or produce any type of hazardous effect. So that is absolutely out of our control. But to prevent that, we can perform off-target binding studies of the molecule with the help of computational techniques. And so this is also one of the part of computational technique of target binding. That is a part of protocol which everybody follows. They try to predict whether the molecule can have various side effects so that it should not be recalled after once you put it into the market because of certain things. But there are maybe teratogenic effects or long-term chronic effects, and those one cannot predict basically. So that happens at the long term. So if these are the reasons, then it is out of our control.
Yes, any other question Kumar Pratyush want to ask? I will just enable his mic. Yes, Pratyush sir, you can unmute yourself and ask your question. Pratyush sir. Just a minute.
Hello, sir. Am I audible?
Yes, sir, you're audible. Please ask.
Sir,
Yeah.
it was a very good presentation. Thank you for sharing your content very precisely. And one thing I want to ask regarding is there any kind of difference when we are opting for free license? So what I have experienced when we are working on 1 month free trial, so the server problem comes and the process is too slow. So is that you are just differentiating the two licenses, free and the paid one, and so that just I want to confirm it from my side.
No, absolutely not. It's the same. Even it's the same which we give it to you. It's the same which we give it to the pharma industry also. It's absolutely same. So it's all from the hardware side. Like it is out of our control. As a software guy, I could say like it's a hardware issue. If it is so, we need to look for the hardware.
Okay, sir. Thank you.
Yeah, can I ask one question?
Yeah, principal sir is there. Sir, please.
Good morning, Dr. Prajwal. Nice to see after so long time, and I'm very happy to see your progress. That our department students has reached to the very good place. So I appreciate your hard work. Obviously, you are very talented at that time also in student life. So I'm very happy with your progress. And I enjoy your presentation, although I am not the hardcore chemistry person or the software person, but still I enjoy that one. I have the one query regarding the CAD one, regarding the target validation and identification. So, like generally what happens in the CAD one, the software will design the molecule or new chemical entity with respect to the target one, and then in the chemistry person will do the synthesis, and then this is the traditional way for the small molecule. Okay, so but if we see now the biologics are there, monoclonal antibodies and all this one, so we know the target for the biologics. But can it possible that with the help of the CAD software to design the biologics, the monoclonal antibody with respect to that specific one? And second question is it easier to make the small molecule or easier to make the biologics with the help of the CAD?
So answering to first question, yes, we do have tools for biologics modeling. You can design antibodies to specific antigen. You can even design a new antigen for your specific antibody to trigger the target, to trigger the immunological response, just like vaccine development. So all type of modules are there. It is perfectly feasible to do that with the help of software. And second question is which one is easy? So I just remember because you said which one is easy. I remember when once I communicated the paper and reviewer replied me back in drug discovery. Remember the thing, nothing is easy. So only thing we can work on making it easier, but it is not at all easy. So, coming towards the question, biologics design is really tricky because you need to deal with the big molecule. But identifying a small molecule which can have potential binding to the target, comparatively, it is easier.
Yeah, correct. Agree. Thank
Because
you.
everything is defined in terms of small molecule. You have definite binding pocket, definite shape, definite interaction. But when it comes to the biologics, it's a huge moiety you need to be.
Yes, thank you.
Next question is from Sonali Walmare ma'am. Ma'am, please unmute yourself and ask your question.
Hello. Hello.
Yeah, madam. You are audible. Please go ahead, madam.
Yes, sir. Sir, my question is as we are discussing about different advantages of CAD and in terms of time, it takes a minimum time for drug discovery, isn't it? So I just would like to know, sir, what are the online platforms where we can see regarding discovery of any new entity which is purely discovered, means purely on the basis of CAD only. It has been discovered as a good potent entity. And
Yep.
would you please explain an example of that in recent years?
Okay, so to my knowledge, there are papers which basically reviews all such molecules which has gone through the process of CAD and how they have gone through that process, which protocol they follow, particularly in terms of CAD. So there are papers. I have those papers, like those are even from recent. Like I have been following such papers from my academic days. So I have those. Maybe if you want, I think that's the best source of knowing like which molecule are interesting, going through
Ye-
CAD, what
oh,
method they are following. You just write to me and I'll share those papers.
okay, sir. Sir, actually, I would like to know online platforms like such some websites or any platform where we can see the list of like, for example, for different journals we are looking for scopus
Mhm.
like that only.
So
I
I
hope you
think,
are getting my question.
yeah, I got the point. You want some website where which consists of all the comprehensive list of all those molecules. So it is really difficult. We need to, if
Not
you
as such
simply,
comprehensive, we can say, but some updates we can get regarding new entities which are discovered or in process on the basis of pure on CAD, we can say.
yeah, I mean, pure on CAD, you need to look into
Or
the
up till now,
website of.
any such in recent some years, any such a drug has been discovered. Means, would you put some light on that?
Yes, so just go to Schrodinger website, you will find list of targets, list of projects which they are working on, and all are based on purely CAD because it's Schrodinger's thing. So you can look into more detail of those molecules. They have also explained which protocol they followed, the papers associated with that. So I expect like similar thing can be present on other company websites, but the only thing is because it's from Schrodinger, so I can be sure like it is purely based on CAD. But if you find something on Pfizer's website, so I am not sure if they follow purely
Okay,
CAD or not.
okay. Just
But nowadays,
company-wise
every
data
Excuse me,
will
molecule
be
sir.
goes
available.
through this technique.
With your kind
Okay,
permission,
thank
may
you.
I suggest one thing to her? So yesterday, Dr. Alex also given his Google Scholar account, and I
Okay.
gone through that account. No, I find various papers where he has given such a molecules which were almost using CAD only to give the molecules and given the like timeline also. So
Okay.
maybe that will be beneficial to her. Just that I wanted. Thank you.
Yeah, that's
Thank you,
great.
sir.
Maybe you can also share that information with me. I'd be happy
So,
to see that.
with your
Thank
kind
you.
question, Dr. Prajwal, I would like to add a few points here. As per the question of Sonali madam, only two companies are now producing the drug candidates through AI, which are in higher stages, as Dr. Gandhi told. Right now, one molecule by AI-driven technology is in clinical trial, developed by a team of Alex, that is in silico medicine. Another company which is creating headline is Exscientia, that is a biotech AI firm. 4 molecule of the company is in different stages of clinical trial. Let us update for the information of Madam also. They are purely through AI-driven drug discovery platforms.
Okay, okay. Thank you very much, sir.
Yeah, thank you.
Is anyone else want to ask any question?
Yes,
Please
sir.
raise your hand. The raise hand option has been given at top right corner of your screen near the chat box. Any question? Please raise your hand.
Bhavane sir,
Yes, sir.
am I audible?
Yes, sir. You are audible.
Sir, there is one question. Thank you, Dr. Prajwal sir, for delivering such a intellectual talk. There is one question, sir. How one can perform post-MD simulation by using Desmos, and how it can get benefited for drug design?
Yes, with the help of MD simulation, you can see it on real time scale, that how it is behaving inside your experimental setup, and how to perform it is like there are few steps one need to follow. It's perfectly feasible. If you want more information on it, I think just write it to me. I will arrange a session for you on how to do it. Like it's a pretty straightforward. You just need to click few buttons and it will be done.
Thank you, sir. Thanks a lot.
Bhavane sir, can I ask one question?
Yes, sir. Why not?
Dr. Prajwal, there are two things going in the market. One is classical QSAR, another one is inverse or reverse QSAR. And basically AI is focusing on inverse or reverse QSAR only. The classical way goes like we have some molecules and we have to find their physical chemical properties or biological activity. In case of AI, we have to give directly the desired property, and we tell the computer give the molecule with so and so physical chemical property. That is, you know better, that is inverse QSAR. So such type of options are available in your Schrodinger application.
No, that's really a good thing to know, and I guess the Extensia in silico medicines they are working on such because they get a molecule as output. But right now we don't have such thing. There are certain reasons behind it.
Okay, okay. And sir, last point from my side. If I say few year down the line, drug discovery will be everyone's cup of tea. Am I okay or am I exaggerating it?
I think a few years down the line, yes.
Everybody means I mean from pharma domain, obviously from pharma domain.
Yep, I would think the same because when I started doing pharmacoinformatics in 2008, that was only one department from NIPER who were working on it. But eventually, with the I think 2008 to 2018, like 10 years of time, I can see almost everybody in the pharma industry that belongs to pharmaceutics, pharmacology, pharmaceutical chemistry, everybody.
Yeah.
Everybody started using these techniques, and this is the way it becomes popular as well as usual. And I second your thought. I
Okay.
agree to that.
Thank you. Thank you. Any other question, Bhavane sir, Wadekar sir?
No, sir.
No.
Because Dr. Prajwal, as one infographics is mentioned on the website of In Silico Medicine. Very interesting infographics they have designed. The left side of the infographic says that to discover drug is like finding a needle in the haystack. And the right side shows through AI you can customize your own needles, generating your own needles. So why to search one, customize and generate many needles. So this all possible because of quantum computers and I think reverse or inverse type of QSAR.
That's really great idea. I think I'll also take a look. It's good to have that slide on my presentation.
Right, sir. Thank you. Thank you.
Thanks for that information.
Thank you, sir. Sir, can we give the concluding remark? Sir?
Yes, sir.
Yes.
If no questions are there, we can process further.
I think one hand has been raised for that.
Yes. Iqbal is.
Wow. Okay, just a minute. I will unmute him. Yes, Iqbal sir, ask your question.
Iqbal sir, are we audible? Now said Iqbal. Yeah, please process. Please go ahead. Or else you can type your question. We will read on your behalf if you are finding any problem in going oral.
Hello.
I think he's having some problem at his end. Yes, yes, you're audible. Iqbal sir, please proceed.
Sir, sir, this is Nausat Iqbal from West Bengal. Sir, I am the pharma graduate, and right now I'm switching my career in MSc in computational system biology and bioinformatics instead of M Pharma. So, sir, this is the right path I am in.
Yes, to be honest, yes, you are on right path. As per my understanding to the field, India has really emerging opportunities. It's a growing field in India. So, I mean, it's a perfect time to switch to such a field.
Sir, you told that you are belongs from pharmacoinformatics from Naipar Mohali, and I also gone through your LinkedIn profile, sir. So, sir, what's the difference between sir bioinformatics and pharmacoinformatics, the subject, sir?
Yes, it's a huge difference actually at technical level. So, bioinformatics, we mostly talk about very basics of sequence analysis and all related stuff. That's
Yes, sir.
at very basic. But when it comes to pharmacoinformatics, we only talk about drugs, drug actions, drug designing. But again, in the background, we use bioinformatics too. So, what will be the difference between pharmacoinformatics and bioinformatics is my programming background is less than the bioinformatics guy. So, he will
Yes,
have
sir.
more programming, and I have less, but I have more application point. So, that's the difference, I guess.
Sir, this is a opportunity in working in a pharma domain.
Yes, absolutely.
Thank you, sir. Thank you, sir.
I think there is no further question from the participants. Okay, thank you very much, sir.
Yep, great. Thanks for the opportunity. I hope to see you sometime in future once these restrictions got over and our travel starts. I will definitely visit Dhule to meet all of you.
Sure, sure. We'll,
We
yeah,
will
definitely,
definitely.
whenever possible, we are very happy that you will visit our institute and we will interact mutually.
Sure.
Thank you very much for the wonderful presentation, sir. Sir has given the insights on Schrodinger software suites in biologics, small molecule drug discovery, material science, and discovery informatics. He has also given the emphasis on molecular modeling, binding site analysis of molecule, core hopping, energy analysis, etc. Sir also has given insights on molecular dynamics simulation, real-time protein and ligand binding using computer. The best part of the presentation was the analogies he has given regarding the astrologist and the QSAR. That was really interesting. As well as the one more analogy has been given, that is descriptor dimensional in movie. That makes us easy to understand the concept in the simplest manner. He has also given insights on the auto QSAR, automatic technique, to show all the possible combination for the future directions to get the lead molecule. He has also given integration of AI, machine learning and deep learning and how they are useful in the drug discovery. Thank you, sir. Now, we would like to present a certificate to sir for this wonderful presentation. May I now request Dr. Raju Wadekar sir to present a certificate of appreciation on screen. Please accept it, sir.
Thank you very much. Thank you.
Thank you. Participants are requested to be here because we are about to start the next session. That is the second session on topic accelerating de novo drugs design using deep learning, by Dr. Arjit Roy.
Yep. Coming towards that next presentation, I would like to attend that one. So, is it
Sure,
okay
sir.
if I be here
Sure,
for the
Yes,
sure.
next one?
definitely.
Okay, thank you very much.
Participant, just wait for 2 minutes. Sir is about to join the session.
Encoder don't go anywhere.
Arjit sir is about to join within 5 minutes. Participants are requested to be there in the session. As soon as the sir joins the session, we will start our second session. It's a request to the participant be remain in the session. Sir, welcome. Arjit sir, welcome.
Can you hear me?
Yes, yes, you are.
Okay, yeah.
Yes, sir.
Prajwal sir, shall we proceed?
We should wait for, I think, 5 minutes.
Yeah, we can start at 12. I mean, if someone has scheduled at 12, I think.
Yes. Sir. How are you, sir? You are fine?
Yeah, I'm fine.
Okay. Just we have finished session of Dr. Prajwal Nanikar from Schrodinger.
Yeah, I was there. And in the meantime, I just wait for 5 minutes. I went out.
Yeah.
Yeah, I was there.
Sir is also there with us. Prajwal sir is also in the session, sir.
Yeah, good.
So, sir, we have with us our respected principal, Dr. Samir Goyal, sir, principal of SVKM IOP, that is Institute of Pharmacy, Dhule. I am Dr. Amit Rangwal, and we have organizing committee members, Dr. Prajwal Nanikar and Pradeep Bhavnesar is there.
Yes, good afternoon.
Good morning, sir.
Yeah. And so I was present, so I know all of you by name. So
Welcome, sir.
thank you. Thank you for inviting me.
Thank you, sir. It's a pleasure, sir. Thank you.
Shall we start, sir? Amit sir?
We can wait for 5 at 12 because some people may join at 12.
Okay, no problem, sir. No
At least 5 minutes.
Okay.
Let me just try to see whether I can share my screen. Can you see my screen?
Yes, sir. Yes. It is
Okay, great.
visible, sir.
Thank you.
So, Ajit Roy, sir, am I audible? Am I audible?
Yeah, you were audible. Sorry, I was on mute.
So, in meantime, I would take the opportunity to update you regarding our campus. Actually, the parent organization is SUKM, the prestigious Sri Vile Parle Kalavani Mandals Institute is our parent organization. We are in Dhule basically, and we have the state-of-the-art world-class infrastructure facility. We are running courses like pharmacy at different level, UG, diploma, and we are also planning for PG level as well. We have engineering campus, College of Pharmacy, College of Commerce and Law also. And on a very aggressive and inorganic manner, we are moving on to make the institute a hub for AI training and research center. So it will be our pleasure if you could spare time to visit personally here and guide us someday as per your busy schedule. that will be kind of you. You want the expertise and incorporation of more and more people who are in the hem, who are the first hand doer of the information.
Sure. I mean, we can discuss all those things. And regarding the institute, I saw some of the pictures in Google, so they are quite impressive.
Right, sir. And as per the guidance and motivation of our principal, sir, recently we got support from NVIDIA also. They provided us world-class GPU as collaborative work also. So we are also good on that front also.
Okay, good. Good to know.
Am I audible, sir?
Yes, sir. Please go ahead, Bhavna sir.
Yeah, thank you.
Roy sir, we are now starting.
Yeah.
Principal sir, we are now starting.
Yes, sir.
Okay, sir. Thank you, sir.
Okay. For second session on topic accelerating de novo drug design using deep learning, we have with us Dr. Arjit Roy sir, Senior Scientist TCS Hyderabad. May I now request Dr. Prajwal Nanikar sir to kindly introduce Dr. Arjit Roy sir.
So am I audible?
Yes sir, you are audible.
Good morning, good afternoon. I am fortunate and privileged to introduce our eminent speaker of the session, respected Dr. Arjit Roy sir. Arjit Roy sir studied chemistry at the University of Calcutta, where he received his undergraduate and postgraduate degree in 2001 and 2003, respectively. He received PhD degree in chemistry from Indian Institute of Technology, Kharagpur in 2009. From 2009 to mid-2011, he was a research associate at the Institut de Biologie Structurelle, Grenoble, France. Between mid-2011 to 2014, he worked as a research associate at Stony Brook University, New York, USA. After that, Arjit sir joined TCS Research, where he is currently working as a senior scientist. Arjit sir has research interests in diverse topics of computational biology and bioinformatics. It includes development of computer simulation methods to understand rare conformational change of biomolecules, development of network-based approaches to understand disease mechanisms, the knowledge of which can be useful for drug repurposing and development of various machine learning methods for design of novel drug molecules. He has published more than 26 articles in well-reputed journals, 3 patents to its credits, and participated in various conferences. With this brief introduction, may I now request Dr. Arjit sir to deliver his talk. Thank you, sir.
Thank you for kind introduction. Let me try to see if I can project it again. Whether you can see my screen?
Yes, sir.
Okay, great. Yeah, thank you again, and thank you, Professor Amit and SVKM Institute of Pharmacy for giving me this opportunity. So we are hearing talk about AI on drug discovery. So during the last few years, we have seen some initial proof of concept on how this method can impact the drug discovery. So many people are hopeful. Some people has also some doubt. So today I will show with some of the examples that so how this method is developing can be useful over traditional methods. And the title of my talk is De Novo Design of Novel Drug Molecules Using Artificial Intelligence. So this is the mandatory slide which shows what are the limitations of the current approach. So drug discovery usually starts from a well-derived hypothesis where first one identify the try to understand the disease mechanism, then from that try to identify the drug target, and once the drug target is identified, then people try to identify drug against them or try to design them. And if you are trying to identify drugs, then you can use something like high throughput screening or even virtual screening that Prajwal showed in the previous session. So then you get some molecules, and gradually that number decreases. The number of molecules that you find during the heat identification step, that number gradually reduces because these molecules need to go through various property checks. So it can be physicochemical property, various toxicity profile, drug toxicity check, pharmacokinetics, etc., etc. And one also need to say that whether it can have any polypharmacology effect or any other side effect. So finally, you can get one approved drug if you are lucky only. But it may happen that you need to start from again. So that's why drug discovery is a long, expensive, and often unsuccessful process. And someone has described drug discovery as a high-dimensional science which may show nonlinearity with many known unknowns and unknown unknowns. In many cases, it can't be defined based on a finite set of rules. So definitely we need new methods. So I will come about that in the later stage. And now let us see what are the current computational approaches, possible current computational approaches. So one can consider as computer simulations. Against Dr. Prajwal talked about that during his presentation. So advantage is that over there you can accurately estimate thermodynamics and kinetics associated with drug target recognition and binding. But the problem is that this is computationally expensive. And mostly you can consider only limited number of molecules here. A viable alternative is the virtual screening, where one can screen a big library of small molecule, and then they need to synthesize, they can synthesize them. And sometimes such already synthesized molecule can also be available from some databases, for example, zinc. This approach is, of course, much faster than the wet lab approaches, which is more resource intensive approach. But here also there is not enough control on the physicochemical properties. Only people look at the target specificity, which is also in terms of docking or any pharmacophore based approaches. One problem is that these libraries are only limited to million molecules, maximum billion. But even if you put some filters on those billion molecules, it will drastically come down. Effectively, the number of molecule theoretically one can synthesize, that number is within 10 to the power 30 to 10 to the power 60, which is huge. And no one has explored that many molecules. So if we can explore that, we can find new diverse molecules which which does not exist in these libraries. So as I mentioned, that this molecule is only a few billions, and after if you put various filters, it can come down to million only. So we need a method that can efficiently explore this chemical space. And at the same time, we also need control of the various properties. So this can be physicochemical property, various toxicity property, and various other late stage properties that people consider, so that we can obtain molecules with the desired properties. Now, let us come to a little bit different point with which I will come to AI in details. So you have seen that during the last few decades, biology has produced a lot of data. For example, if you just consider the Human Genome Project after its completion, so there was a huge amount of data. Even if you look at the various multi-omics data, since now the methods are cheap, you can generate a huge amount of data. If you only look at the drug space, now there are data on the drug-induced gene expression data. Now there are side effect data. You can see the various failed, experiment on those failed drug. So those data are also available with pharma. They have freed some of those data. So these data needs to be effectively stored and digitized. But these data is useless unless we can get information out of it, unless we extract knowledge out of it. And who can better do that job than AI-based method? There are some problem over there. For example, the data reproducibility can be challenging. The quality of the data can be challenging. But if you can overcome those problems, the big data and AI can create number of important things. And it has already started impacting various fields of drug discovery and development. So today we'll focus on the design of novel small molecules, some property prediction, and the structure activity relationship mostly. But there are also other areas. For example, one can use the text mining to find out from literature some information, and together with the multi-omics data, one can find the relationship between the disease and generate some hypothesis, which is also another important part. In the later stage, when I will talk about the TCS drug discovery platform, I will little bit touch about this point. There are others area, for example, selection of, how to select patient during the clinical trial management. TCS also platform which now many pharma companies use. There are other areas, for example, automatic document review for approval, detect adverse and safety issue throughout the later stages after the drug is approved. Now, the AI-based drug discovery, there were some attempt during between 2013 to 2015, but at that time there were not much success. Maybe because the method were not developed, the issue of data was also there. So by 2018, we could see that there were almost 250 startups were working in this field. In 2018, we saw a number of important papers. So one important paper was from Popova et al., which was published in Science Advances. I have references of it later. And then in 2019, we heard the talk by Alex yesterday. So we saw this paper from Nature Biotechnology in Silico Medicine published this particular paper. And in February 2020, we saw a paper from MIT which talked about the antibiotic known as halicin, antibiotic discovery. This particular antibiotic was known as halicin. Then BBC reported that how artificial intelligence created medicine went for the clinical trial within 15 months, which is remarkable. Although in this particular work they didn't describe exactly what method they used. So, and since then there were many method came up, many paper came up, and we have also contributed. Now, before going into that, let us see what is artificial intelligence. So artificial intelligence can be described as intelligence demonstrated by machine as opposed to natural intelligence displayed by humans. So the most important part is that AI technologies can extract concept and relationship from data and learn independently from data pattern. So in my first slide, I mentioned about the many known, unknown, and unknown. So maybe it can happen that machine can learn this data pattern which is not obvious by human. And we are particularly interested about the deep learning methods, which utilizes a logic structure similar to the brain called neural network to recognize and discriminate pattern such as speech, image, and video. Now, these deep learning methods originally developed for computer vision problems are now delivering almost near human accuracy. There are a number of application areas that we are already aware. For example, facial recognition, we can see them in our mobile or other secure places. There are cases of augmented reality. In health, by health here I mean that those images like X-ray or MRI, where one can say about the presence of disease or not, and also self-driving cars. And one such example is in the left-hand side, where you can see that the method can detect multiple objects, and at the same time, it can discriminate between them. So it can annotate image. Now, it can also generate new images. For example, given a picture, it can make it smile. It can tell what will be your, how will you look after a few years. It can change gender, etc., etc. Although these are all toy example, but this can be very useful. Because what we want to explore the huge chemical space at the first bit. We need to generate new smiles. So the same concept can be used. So there are a number of new methods that can be used here. So Dr. Alex yesterday talked about a method. So what we are interested is that we are interested about the autoencoder or variational autoencoder. I will come in the next slide. So what is the autoencoder? So autoencoders are an unsupervised way of learning the lower dimensional feature representation. And this important part is that this feature can be learned by itself. They are comprised of two connected network. One is called as an encoder, another is called as a decoder. Now the aim of this encoder is to take an input and produce features out of it. In the next part, one can think of reconstructing this input data, and for that purpose there is a loss function, and this loss function we need to minimize this loss function so that we can reconstruct the input data, something close to it. Now, what is variational autoencoder? So in case of a variational autoencoder, it models the probability distribution of the training data. And from encoder, there are two output vector. One is the vector of the mean and another is a vector of standard deviation. Now, what does this mean and standard deviation, what is their role? Now, the mean controls the position where the encoding of input should be centered around, and the standard deviation controls the how much the mean encoding can vary. And there is also a loss function that similar to here, which is known as the Kullback-Leibler divergence, which measures the how different two probability distributions are. And similar to the previous case, one need to minimize the Kehl divergence. Now, in order to generate new image or even text, we can have a random we can have a probability distribution with some mean and standard deviation, and from that we can generate new output. Now, how it works for SMILE or the small molecule? A small molecule. So a small molecule can be represented in a various way. So one way of representation of it is 3D. Another way of representation is 2D. So I will, so we also frequently use 2D, but today what I will talk about is 1D. So the 1D representations are known as SMILES or Simplified Molecular Input Line Entry System. So many of you, I'm sure most of you are aware of that. So a SMILE also has a well-defined grammar. So where is the double bond, where is the single bond, if it is an aromatic ring, how it looks. So what happens in the variational autoencoder model? So we supply a batch of SMILES, and what the model does, so model try to learn various features and produce the after parameterization, the mean and the standard deviation out of it. So basically it is trying to learn various features. For example, what are the number of carbon, what are the number of nitrogen, how many double bonds are there, how many aromatic rings are there, how many nitrogen or oxygen in the aromatic ring. Overall, how the molecules or the SMILES look. And because its job will be to reconstruct or generate new SMILES, and for that purpose it tried to understand how it can pick up atom after atom or character after character when it generates SMILES. So that's how we can have a generative model. So this generative model learn the grammar of small molecules and design novel and valid small molecules. Then also we need to have a property model, a predictive model which can predict the property. So these predictive model are nothing new. It's similar to the quantitative structure activity relationship model, which learns the mapping between the small molecules, here represented as SMILE string, and then corresponding experimentally determined property value. So this, so only difference is that here we are using SMILES as descriptor. But there are various other methods which are much more advanced. For example, graph-based method, where this type of so we approach the problem in a different way, and we have also used them for property prediction. So I will not go into those details today. So next, what happened? So we are generating new molecules using a generative model, and with the introduction of the reinforcement learning, we are generating new molecule, then we are passing it to our predictive model. So now predictive model can be for, let's say, target specificity. It is either calculating IC50 or some docking score or some physicochemical property. Now, if that particular SMILE has the desired property, then a reward is given to the generative model. Otherwise, the model is penalized. So this is a big advantage over the traditional method, and this way we are optimizing that property so that the during design process itself, it can learn the particular property. Now you can consider a single property. You can I will come about the multiple property during the later stages of the model. So there are a number of advantages here. So next, I want to introduce another concept which is known as the transfer learning. So what we did during the generative model, we have we started from a public database through which, I mean, whatever knowledge we have on smiles, we train our model with that data of the small molecule which is represented as smiles, so that our molecule can learn the grammar and it can explore the vast chemical space which is colored in blue in the left hand side picture of the model 1. Now it might happen you are designing molecule against a particular drug target, and we already have some knowledge, like some structure activity relationship knowledge. I mean, some of your fragments or molecules are showing some activity. I mean, it's not important whether they are good or bad. Even if they are bad, but they are showing minimal activity, that is fine. So you can incorporate that domain knowledge into the model. So this concept is known as the transfer learning, so that the model can also, instead of focusing on everything, it can learn the feature of that data, but it also retains the diversity already. So the rest of my talk is organized as below. First, I will talk about the ligand-based drug design. I mentioned about that multiple properties need to be considered during the drug design. So I will talk next about how we can consider multiple parameters during the drug design. I will talk about how we can incorporate explainability in the model, because some people say that the deep learning models are black boxes, how we can incorporate explainability over there. Next, I will talk about the structure-based drug design. Then I will little bit talk about the drug discovery, the drug design platform that we are developing, or we have partly developed at the end. So this is how our ligand-based ligand-based drug design pipeline works. So I already have mentioned more or less all these things. So we have a generative model, and we also have a predictive model which predict the property of these molecules. We do this on the fly property optimization. Sorry, using reinforcement learning. And if you have some domain knowledge about the target specific data set, then you can use that. So today, I mean, our model can also do some additional things. For example, we have shown that this method can also be applied against novel proteins where no such target specific small molecule data set is available. So today I will talk about that part, and we can do that using evolutionary information. So this is first, what is the performance of our pre-trained generative model, and for that purpose we looked at checked our model with a benchmarking data set known as the guacamole data set. We found that our method is producing good number of chemically valid molecules which we tested with RDKit, and they have high internal diversity. That was another thing that I have already explained to you. And the other thing, we need to check that we are generating new small molecules, but if they does not satisfy the various drug-like properties which make the small molecules a drug, then it's of no use. So we also check that whether they have the desired drug-like properties, and various things can be checked over there, like log P, molecular weight, number of hydrogen bond donor acceptor. QED is a estimate of various physicochemical properties, and also we see that if they can be easily synthesizable. So next we applied our method against the Janus kinase 2 or JAK2, which is a part of the JAK-STAT pathway, and it's implicated for various immune responses. So for this particular case, we assume that JAK2 is a novel protein, and in order to do that, we collected all the known inhibitors of JAK2 and we completely separated it from our model. We just kept it aside because since we assumed that it's a novel protein, we kept all the known inhibitors in a separate place so that later we can validate with whatever we will generate. So instead we collected all the inhibitors of homolog of this protein, and that was used to train our generative model. We know that that data is not perfect, but in the subsequent slides I will show that how the reinforcement learning and the model will force this data set towards the towards more JAK2 specific molecules, towards the creation of more JAK2 specific molecules. Now this collecting of this data can be done in a multiple way, and we have multiple proof of principle for this. So next we generated number of molecules, and we saw that some good number of molecules has a good Tanimoto coefficient with respect to our validation data set. So what is Tanimoto coefficient? Tanimoto coefficient is the with Tanimoto coefficient we measure the similarity between pair of molecules. So whatever we generated, we compare it with the validation data set. Now let us look at the figure at the right hand side. Since we assume that we don't have any known data about no knowledge about the inhibitor of the genus kinase 2, we had, in order to see the target specificity, we had a docking score based model. So when we try to generate new molecules specific to JAK2, we optimize the docking score because we have no other option. Otherwise, we could have a IC50 based model. Now, what we can see from this figure B is that the red one is the predicted docking score for the training data set. Now, during the after reinforcement learning, which is shown in blue, that the model could produce molecules with the better docking score. And next, I will show you that how they are closer to the how they became specific to JAK2. And in the meantime, we also checked whether this molecule possess drug-like properties. We also checked the synthetic accessibility score, which is good. I mean, that we can see from these results. So now how we can say that our model during the reinforcement learning step, the method could shift the training data set more towards the validation data set. And for that purpose, we collected the top 10 fragments from the validation data set. So they are numbered as 1 to 10. Now for this validation data set, we try to see the frequency of them during the each step of the model. Now if you just see at the last 4 columns, you can see that during the after the model, after reinforcement learning, these numbers are more close towards the validation data set. It's not the same. It's not possible to be same, but the number move more towards the validation data set from training data set. Next, we also see that this our optimized generative model that we obtain after reinforcement learning learn from the training data set and produce molecule close to the validation data set. So if you just look at the lower 2 figure, you can also see other figure, but at the moment, if you can just look at the lower 1 figure, maybe for this molecule 7386, which we have generated, we could see that there are certain molecules which are present in the validation data set and the training data set. The corresponding ID in the validation data set is shown in green, and the one in blue is shown in is from the training data set, and in the bracket are the corresponding Tanimoto coefficient. So what we could see that here in this particular case, it could learn from the training data set, which has a much lower Tanimoto coefficient similarity, and it could produce molecules close to the validation data set where it has a high Tanimoto coefficient. So at the moment, I'm mostly describing about the similarity because I have also a challenge to show that our method is it's producing something very similar to the validation data set. So that's why I'm talking more about the similarity. But at the same time, we could produce new scaffolds. We show in this particular paper that at the 25% of these scaffolds are completely new that we have generated. Oh, and they are also with high docking score. We could also produce 3 molecules which are identical to the existing JAK2 inhibitor. Then we also see which were the functional groups that make the molecules selective towards JAK2. And for that purpose, we went back to literature, to the patent document, and find out some of the functional group which make molecules selective towards JAK2. And we saw that our molecules also possess those functional groups. So when we submitted this article in towards the middle of 2020, we don't had a method which can calculate selectivity. So in the next part, I will talk about this selectivity, which we have, which can think about this, say more about this selectivity, because this is a big problem. It can lead to off-target side effect in the subsequent slides. And also, this slide shows that 2 molecules we saw that they are highly selective to JAK2 over their homologous protein. So they are shown in the box in the left hand side, and we could see that we could generate molecules very similar to them. So 2 for each cases are shown in this figure. And what is important that we also could see that some of these molecules are lies at the very top of our priority list that we have produced. So this gives us more confidence. So this is a more recent work that we have done. So I was talking about the question of selectivity, and at that time we had no method which can calculate this selectivity. So this is a more we used a multitask deep learning model for this. So what is the problem statement here? So the problem statement is that often you create hit molecules which is originally intended against a particular protein. So let's say in this case, this particular protein, protein 4. But so initially you show activity against this protein 4. You didn't care about the other proteins. So, but later on you can see that your hit molecule is not only inhibiting the protein 4, but it can inhibit the other proteins also, especially if they share the sequence similarity or some sort of functional sequence similarity. So that's why we need a selectivity model, and this also makes sense, because if you look at the total human proteins which are druggable target, if this number is within 660 to 700, about 44% are from homologous protein families. So here we focused on a more shorter problem on just looking at the homologous protein family. We just created the proof of the principle based on 2 protein family known as the JAK family and the dopamine receptor family. But this can be extended to kinase profiling or for other cases through which we can understand the off-target side effect, the effect which is more popularly known as the polypharmacology effect. And these are the performance of this model. So we used at least 5 methods here, and this is more or less their performance. So I'm not going into that details. But what is interesting here is that we also could identify the functional groups which make each of the protein of this family selective towards, so which functional groups make them selective. So that also we identified through the explainability model. So we recently put this article in the preprint server, so you can check out if you want to know more about this. So we also did a similar work on SARS coronavirus 2, where we identified few small molecules against the 3CL protease due to short. So I will talk about much more other things today. So these also used a similar approach. So I will skip this. But if you are interested, you can look at this particular paper. So next I will come to the problem of multi-parameter optimization. So I mentioned about this problem during the initial, during my first slide, that multiple parameters needs to be checked during the drug design. So at the initial stage, people mostly consider the bioactivity of the hits that they have identified. But subsequently, various physicochemical and pharmacokinetic properties need to be optimized. So that's why those numbers gradually decreases, because a small molecule can be a good drug only if it also possesses a good physicochemical and toxicity profile. Otherwise, this can lead to uh, that's the reason why there is a high attrition rate during the later stage of drug design if we don't care about this particular problem. So we asked the question, can AI-based method can reduce this design cycle so that you don't need to start it from very beginning again? And this is so now what we are doing additionally here is that we are not only looking at the target specificity, we are including number of other properties during the initial stage of drug design itself. So for that purpose, we have taken a target protein from the central nervous system, which is known as 5-HT1B. And as many of you know that for any target of central nervous system, many physicochemical properties influence its ADME, binding efficiency, and the safety. And apart from the target specificity, one also need to look at a protective barrier known as the blood-brain barrier. And one need to check whether the new molecules that one is designing that can cross this protective barrier. So we checked a number of physicochemical properties which influence the overall safety for any central nervous system drug. For example, partition coefficient. Then we also looked at the topological polar surface area and other things. So molecular weight also need to be usually lower. So we consider a number of things, and at the end we consider only 4 of these property: the target specificity in terms of docking score, the blood-brain barrier predictive model, a partition coefficient predictive model, and also we consider molecular weight. And then we started designing molecule. We produced few molecules. We found that a good number of molecules were similar to the validation data set. Here also we separated the validation data set so that we can validate our method. So that one interesting thing that we did here is that we try to find out the contribution of each of these property that were used towards the molecular design. And what we found out of that some of these properties can be conflicting. So what particular property need to be optimized, that's also you need to be careful over there. And we also see that some of them also can support each other. So that's why there were requirement of 4 properties over there. And the picture in the left hand side shows the property distribution from the known BBB permeable molecules and the molecules we generated. There are some difference, but we could overall cover that property space. Next, I will come to the question of explainability, because when we are giving some molecule to a medicinal chemist, if we can tell them that why my molecule, for example, can cross the BBB blood-brain barrier, or why it has a certain toxicity value, which functional group has a responsibility for that, then it will be very good for that will increase more confidence of a medicinal chemist on our method. So this is something very similar to what Prajwal showed in his slides. I mean, we could also, from the feature attribution method, we could also say that what are the functional group that are responsible for BBB permeability or BBB impermeability, and then later, and we color them. If they had a positive feature, we color them as red. If it has a negative feature, we color them as swan. And then we went back to the literature and tried to see that whether our predictions is right. And another example is shown in the right hand side, where in the middle there is a molecule which is a known BBB permeable molecule. So why we have considered it? Because in our generated set of molecules, we could see number of molecules very similar to it, but different BBB permeability score, and we could also use the same method. The coloring thing is not shown here, and we could also explain which functional group is responsible for BBB permeability or impermeability. So we are learning a lot from this. We are incorporating explainabilities to the so-called black boxes. So next, I will talk about our receptor-based drug design board. So till now, whatever I have spoken is mostly regarding the ligand-based drug design, where an initial target-specific ligand data set is necessary. We also showed that how it can be applied for novel protein by using a different approach, but this can still be a challenge, especially for some novel protein or some orphan targets. And even if you go into literature, so most of these AI-based methods are ligand-based. So we thought of that if we have only a protein structure similar to the whatever domain knowledge we have, so can we create molecules based on the protein structure alone? So we asked that question. And so we also have a method, so I will go into that in the next slide. And for that purpose, we collected data from PDB bind and scPDB, which gave us various protein ligand complexes. So there are also bioactivity value in different forms like IC50Ki and KD. We collected all of them because our plan also is to create a bioactivity model out of it. And this is the model that we have. So first we look at the protein active site, and we model it based on a graph-based approach, where the nodes are the amino acid residue, and if they are close enough, we put a age between them. We also put some additional feature over there, for example, whether they are hydrogen bond donor, acceptor, their polarity, their volume, etc. So that and we used a graph variational autoencoder approach where we can, so that the model first learn about the active site structure, and we also have a smile generator. Then we merge the Latin representation of the graph-based method and the smile-based method, and then under the guidance of the DTA model within the reinforcement learning framework, again, we started producing new molecules if you give a new active site. So even if you give a new active site, it has already learned from the known protein ligand complexes. Now, if you give a new active site, which is responsible for protein ligand complexes, response where the ligand can bind. If you have that particular active site, then the model can produce molecules which can fit over there. So this is the performance of the model. I'm not going into those details, which is very good, I would say. And then we started generating molecules. Again, we took two test cases. One is the JAK2 and another is the TRD2. So for the JAK2, we could see that there are 30 molecules which has a which have very high good Tanimoto coefficient similarity. Some of them are with the known JAK2 inhibitor. But here I just want to say one more thing, that there is some inherent problem about the Tanimoto coefficient based similarity, because if there is a chlorine to change of fluorine, then this Tanimoto coefficient will decrease, although they possess the same property. So for that purpose, we took a pharmacophore based approach. So I don't need to repeat it here because Prajwal kind of mentioned about these things in the previous slide. So if you are not there in that talk, so it basically look at the 3D pattern of features responsible for binding that is shared by all or most of the input ligands. So that's what the pharmacophore. So instead of a atom to atom similarity, now we are looking at the 3D pattern. And now when we compare our generated molecule with it, we could see that the 90% of our generated molecules have a very high pharmacophore level similarity to the existing inhibitor. So then we approach the same, we applied the same method against the DRD2 target, and here we could find that we could generate 5 molecules which were identical to the known to existing DRD2 inhibitor, and 80 molecules had a high Tanimoto coefficient. We also looked at the fragment distribution for each of these cases and how if they matches with the validation data set, and we also looked at the pharmacophore based approach, which is again good. Next, we tried to understand what exactly the model is doing. I mean, can we find out more information out of it? And for that purpose, we try to find out important residues from the graph attention based model. And the graph, the attention coefficient says about the interaction probability distribution over each node and its neighbor in the graph. And by analyzing this attention coefficient for each node, the residue pairs which are frequently given more attention by the model can be identified. Now, for the DRDT case, we saw that there were almost 17 interactions which had a very high attention coefficient. Now we look closely about this residue which are involved in those attention coefficient, and what we found, there were almost 8 active site residue which has more attention coefficient over others. So next, in order to understand more about this model, we try to see what are the interaction of these residues with the molecules that we have generated. And what we found that some of them interact through hydrophobic or hydrophilic interactions. And already from literature, we know that 3 of them, leucine 94, tryptophan 100 and isoleucine 184, from mutational study, we know that they have role in stabilizing the protein ligand complex. And another interesting case is this ASP114, which is known to anchor the secondary amine group present in the inhibitor. And although we cannot clearly say it, but most likely this graph attention based model, this important residue that we found from this the graph attention based model has some role in guiding the molecule generation. So now I will more or less conclude about this part. So what we have learned about the advantages of the deep learning based approach over the traditional approach is that now we can explore a huge chemical space. We can explore the diversity. Again, I had a liability of proving that my method is working, and so that's why I discussed more about the similarity with the existing molecule. But as I shared earlier also, so we produced diverse molecules scaffold, completely new scaffold, and if you want to see more about them, want to know more about them, you can go back and see some of our paper. And the other thing is that we can do the on-the-fly property optimization. It can be bioactivity, it can be the docking score-based method where there is no information is available. For the structure-based method, also we showed a bioactivity model, and there can be so this on-the-fly property optimization is one of the important advantage over the traditional method, and you can include more than one physicochemical properties when you are designing molecules. You can include various late-stage properties during molecular design. And I also discussed something about the explainability, although we need more improvement over there. So there are some areas where we need more careful consideration. So any AI-based method need more good amount of data, but the quality of the data is also important. So we always need to be careful about that. And I think if pharma companies open up more their legacy data, there will be much more opportunity for this particular field to develop, which can learn from the data. At the same time, we also need to see or develop a method which can work with the low data scenario. Possibly deep learning method like few shot learning type of method can be useful over there. One also need to work more towards the reaction informatics or the retrosynthesis approach. Uh, some groups are already working on that. We are also working on this on the retrosynthesis approach, and I hope that with the advantage of the method, with the advantage of more data, will be this particular field will be more successful in that area. And the biggest part is that we should always try to incorporate more and more domain knowledge into the decision making. That will make our method much more improved. So AI is not here to replace human. It will take the help of human to learn more and learn from those patterns that is not obvious for the people, and try to generate more knowledge out of it. And one also need to understand more from these black boxes, so that there is more confidence on the model. So next, I will talk a little bit about the drug discovery platform that we are building. So what I have discussed, so that's one of the main part which talk about various generative model and the predictive model. Various other features like selectivity, kinase profiling, toxicity profile. I have not discussed much about them. But there are also other part of the overall drug discovery and development. For example, one need to think of a well-derived hypothesis even before you start designing. And for that purpose, network biology approaches can be very useful. One can use text mining, and one can use number of multi-omics data and incorporate into a particular database. Why it is important? Because these data are often scattered in multiple sources. So if we can incorporate into a particular data set, we can identify the entity. We can do the entity exploration between them. So what is the known knowledge between the gene, disease, and drug? Because all these databases give separate information. Now, when you merge that, you can get those information about the gene, disease, and drug relationship. Then you can create a network and visualize them. You can use various network-based approaches for the link prediction, which are originally developed for link prediction, various AI-based method over there to understand the disease mechanism, prioritize genes or proteins, and you can also do the disease try to find the disease relationship. What can be the reason for comorbidity? I mean, how more specifically how one disease can relate. Can enhance the another disease, etc., etc. How there is a drug can affect another disease, and if you can do all those things, probably we can also identify a drug that can be repurposed. And we have done one such work. I just want to give one example here. We have number of different example in this area from different groups of PCS. We have also published some papers. So in this work, we worked on COVID-19, where we could show that what is the relationship between COVID-19 and multiple other diseases. So here is only a small snapshot of that information, and how different pathways are related among those diseases. So it can tell how this can lead to different disease. This was done early during the COVID-19, although the paper was published later. And later, using the drug-induced gene expression data, we tried to repurpose the known drug. And one such drug was COL3, which later went to clinical trial. And this particular drug was from a group from New York known as the CMT-X. We are also working on a number of projects with our academic collaborator on the modeling and simulation part. This is also a important area, and traditionally this was there in the. There are many people in the drug discovery pharmaceutical company who are using this type of approaches. In the last 2 years, we have published a number of paper on this direction. And we are also working on Dr. Reddy's Institute of number of this project. For example, one is the screening of ultra large libraries. And we are also trying to do validation of some of the drug design work with them. So this is how our integrated de novo drug design platform looks like. So some parts are, we have already built, and some part we are building. So we'll soon take it out to customers and people. Generative models are at the heart of this platform. And as an input, one can consider multiple things. If it is a ligand-based data set, one can do the ligand-based drug design. If it is a protein structure alone, target structure alone, then one can use a receptor-based drug design. You can also supply the gene expression, then one can use molecules with the desired gene expression. So this is one of the work that I have not discussed today. And if you don't know anything about your input, you would like to go back and look at the disease, try to understand the disease mechanism, try to identify the protein target, and for that purpose you can take the help of our network biology approach. Then during the generation of molecule, one can use single or multi-property optimization. So the most important part is the bioactivity, of course, and at the same time one can use number of other properties, physicochemical properties, or the late stage properties and one such example that I shown. And then you can calculate the various toxicity as well. I didn't discuss much about them. And if you're still not convinced, you want to check out the various free energy of binding, maybe you'd like to look at the molecular modeling approach that we have. And visualization always remains a big problem for the drug design. So you need to have method which can visualize it. So with this, I would like to end, but before end, this is my acknowledgement slide. So whatever I present for the drug design part, so most of those work has been done by Soumya and Navneet. So Dibbo and Bruto is mostly involved in the network biology part work. The molecular modeling and simulation work has done by Shiladitya and also Navneet. So all of them help each other's project. And I would also like to thank Dr. Gopalakrishnan Bulusu, who is advisor to life sciences research, Dr. Rajgopal Srinivasan, who is head of life science research, Dr. Gautam Shroff, who is head of overall TCS research, and Amit Saxena, who is from the business team and who heads the genomics platform and also responsible for the drug design platform that I discussed. And among the academic, I would also like to thank all my academic collaborators. And among them, with Dr. Reddy's of Institute of Life Science, we are trying to validate some new molecules against some diseases which I have not discussed today. Thank you very much for your kind attention. And if you have any question, I'll be happy to answer them. Thank you.
Thank you, sir.
The session is open for question and answer. Amit sir, please unmute you.
Okay, those who are having question, please raise your hand. I have unmuted Rahul Das, sir. Please ask your question. Unmute yourself and ask your question. Another raise hand I can see over here. Ajay Kumar Gandhi, sir, please ask your question. Unmute yourself and ask your question.
Yes, thank you, sir. So before I ask question, sir, I just want to know whether I understood the thing clearly and rightly or not. So in deep learning, what we do, for example, we have certain enzyme and we have say some 100 molecules, and we try those 100 molecules against that enzyme, and we try to find out, maybe with the Qsar or pharmacophore modeling or maybe docking, try to find out what functionalities, what scaffolds are there which are working for the activity or against the activity. And as you said, that in deep learning what we do is, those who increase the activity, we reward them with the deep learning, and those who are not, those decrease the activity, we are going to penalize them. And of the things, like of those chemical features which are rewarded, among those you do with all the permutations, combinations, you generate new molecules. It is something like scaffold hooping only. And those molecules again will be treated against with that same enzyme, and we again find out the same thing, and we do this, we go on doing this again and again till we reach to the final set of chemical features which are rewarded or which are good for the activity. In this, we have different fragments, and we connect them together after knowing the different fragments. But whether at that time we take into this consideration that they are having proper conformation, configuration, and I mean, when we try them at the beginning for those 100 molecules, we optimize them. Whether that same optimization also we do perform, and is there any contradictory things, like certain fragments we find which are working for, but when they are not in certain conformation or configurations, they are not good, and that's why at the end somehow they get into penalized part of the fragments. That's my the question, sir, whether this is what the deep learning is doing. And
right, you are absolutely right. So there are certain advantages over those methods. So what it does, again, I'm saying that it learn those features and try to produce, it try to focus at that chemical space. I mean, if you have some known active, even if they are not a good active, but a certain activity it shows, we consider them, and then try to learn those features and try to generate molecules with those scaffold, and at the same time with much more diversity. So that's the concept, and at the same time we try to do optimize this property. So that's a big difference over the traditional method. So during the other approaches, you may are looking at one property, but here you are optimizing multiple properties. So that's also a big difference over the other method. And regarding the question of the conformation, so we need to still go a long way regarding that.
Okay, thank you, sir. I have one more question. If organizers permit me, I can ask. Otherwise, they can give chance to other participants.
Yes, please ask.
Actually, sir, my question was, since yesterday this was in my mind to ask you also. Sir, we know the delivery of drug, like when you take the example of a drug that are administered orally, they pass through the physiological system like stomach and intestine, liver, and then cell wall, cell organelles, cell fluids, and all the things. We, to much extent, we know all these environments where through which the drug pass and reach to its target site. Sir, in chemistry, we know the concept of retroanalysis. We know the molecule which we want to synthesize, and in retroanalysis, we just make it synthons and we find proper synthetic equivalents and we do its synthesis. There are certain softwares which are developed. I think Stradinger also developed one, Cambrixis. I am maybe wrong with the name, but that software gives you path also. That is the feasible and best suitable path for the synthesis of drug. The same concept, same approach can we have in the drug designing also. Like for certain enzyme, we have like this with deep learning, we find out the certain molecule is best suited and that inhibit the activity of that enzyme. And now we are detaching that molecule from the enzyme. Now I'm talking retrospectively. Like we detach the molecule, we take it to the cell wall. Now we know what happened at the cell wall and how it can enter. So we have taken it out of the cell wall now. Then we have passed it to the we reach to the through the bloodstream, we reach to the liver, and there also it undergo filtration. There some reactions happen. Now it came to back to alimentary canal, and now the out of the body admissible drug form. So this way, some way can we proceed for the drug designing, maybe not now, but in the future. Or if it is, then any research group that you know that is working in this direction.
So kind of, I mean, I don't know if I have understand your question properly, but what I can say you that if there is any knowledge or data about whether it can cross different barrier or different region, so that's what I said when I talked about the multiple parameter optimization. So over there we consider one such property like the blood-brain barrier. But here also during I mean, whatever you said, this can lead to various toxicity effect. Now, I have seen some data regarding whatever you just mentioned. So these data again can be included in the initial stages of drug design. So that's the advantage of this method.
Yes, sir. You talked about toxicity, sir, and in your slides, you shown one slide where you have multiple enzymes on the vertical side, and in horizontal side, and on vertical sides you tried some drugs, and there you put some tick marks and cross marks there.
Sir, in that particular slide. So over there we considered 4 properties. Now whatever you were saying, some of those can also be included over there, but that will depend case to case. For example, some various toxicity are there. So, and that is dependent on different target proteins which are reside at different area. So accordingly, one can choose such problem. For example, for some cases, if your cardiac toxicity is important, so over there you can include that particular thing when you are designing molecules against that particular target.
Okay. I mean that we have to go in one another dimension also, like not only the molecules against one enzyme, but we have to do all these things against all the enzymes possible which fall in.
That is also possible. That is also possible, and I think we are also working in one such problem. I didn't describe today.
Okay. Thank you so much, sir. And the question that I was asking, no, sir, like when I'm saying that we are taking molecule out of the cell wall, we know which functionalities will help that molecule to enter the cell wall. So while taking outside, we were not with those inside the cell wall, we were not with those functionality on that particular molecule or drug molecule. But when we are taking it outside the cell wall, we will put those functionality on that, and then we will take it through the lab.
Yeah, I think there are some drug which can be modified only on certain condition. I mean, within the body. I'm just forgetting the name. So maybe you are asking something like that, but yeah, I'm not aware. I mean, I know that there some drug exists like that. I'm forgetting the name, but yeah, there are some drugs, but I don't know how that can be approached with a deep learning approach.
Yes, sir. And sir, the paper you told about right now, just I would like to read that paper also, and I would like to know any scientific community that is working in this area or domain regarding.
There are many. I mean, you also heard Dr. Alex spoke yesterday. Maybe they are using some different method, but I think they are also. I mean, there are many number of people are working in this field.
Okay. In India also, sir.
I'm not aware much in India.
Thank you so much, sir. Thank you, organizers.
Prajwal sir is also having one question. Prajwal sir, kindly unmute yourself.
Yes, yeah. Thank you very much. First of all, Dr. Arjit Roy, for such a nice presentation and the description of AI applications in various CAD domains, and it was really fascinating to see it. As I was I am really new in this AI application field. So I have actually three short questions. First one is, maybe I'll just go with one by one. And the first question is, can we quantify the amount of computational time which is required to generate a model? Maybe you can take any of the example out of all. And based on available computational infrastructure, let's say one CPU or one GPU, and then can we somehow find out the timeline? How much time does it take? That's the first.
Yeah, so we do it regularly. I don't know exact the number, but what I can say you that this once you have, so these pre-trained generative models are always pre-trained. It's already available. The same with the predictive model. Now, once this is available, now if you give me today a drug target, I can do everything, including some studying the protein and the understanding about the a little bit about the if I take the structure-based approach a little even after studying for couple of days, I can do the all the work within 2 weeks. So that much faster it can be, and I think. I mean, I don't know the exact number of GPU or CPU is used. We use the GPU cluster that we have locally, but I think it should be usual. I mean, that can be available any institute.
Okay, so that's great. Thank you. And next question is, I was curious to know the molecules which you have presented. So whether are they performing the same way when you put it into the experiments, or it is still stick to the in silico studies only?
Yeah, so that's what I mentioned. So we are trying to validate some of those molecules. We have not tested, and probably with some pharma companies we'll be able to do that soon. That's
okay.
The hope. We are trying to take it forward.
Yeah, that's cool. And about the source code, like, is it a open source available somewhere on GitHub or somewhere? Or it's
yeah, we can discuss more though at offline.
Okay. Okay. So thank you for answering question.
Thanks.
Sir, I have one question. So there are three terminologies: vanilla, autoencoder, standard, and classical. All three are synonyms? Vanilla, standard?
No, all these are different part. I mean, there might be some relationship between them. So actually, I put a particular paper which came out by one of my with whom I worked for a long time, Dr. Devapriya Kumar from IIIT Hyderabad. So he has written a very nice article. It was as one of my, I gave that reference when I talked about the artificial intelligence slide. So they have recently written a very nice review on how these methods, what are the, in a very simple way so that it can be useful for scientific community. It was published in Chemical Science journal. So maybe you can see that particular article because it's very well written. Everyone can understand. So maybe you can, I mean, most of your concept will be clear from that particular document. So I will advise everyone who is interested to know the basic methodology behind that. Because in the paper, we need to focus on the smaller things. So maybe you will not get much details in our publication because of, I mean, apart from whatever we have developed, the new things, the older things we cannot give to publications.
Yes, sir. I think very jumbo article it is. I think if I am not wrong, very big article it is. You're talking about.
Yeah, it's
ACS. I think around 2 years back. I think 2020.
No. I'm talking about a article which just recently came out.
Okay.
2022.
Any questions from your side? Principal sir is with us.
Okay.
Yeah, thank you, sir, for the detailed informative presentation regarding the de novo drug design synthesis using the deep learning. I thank from the SVKM Institute of Pharmacy for sparing the time for the first international webinar series on artificial intelligence in drug discovery. So thank you very much, sir, from this CMI.
Thank you very much for inviting me.
Thank you, sir.
I'm glad to be part of it.
Same here, sir.
Sir, I have a few more questions, if you permit.
Yeah.
Sir, what is the meaning of probability distribution? Exact simplified meaning of probability distribution in terms of sampling for de novo drug designing.
Okay. So here you are trying to learn the various features, and for each feature there is a probability distribution with mean and standard deviation, so that the model is learning the distribution of various features, and it learn so that there is a atom by atom, because at the end what it has to do, it has to create a new molecule or smiles. So there is it try to learn those probability distribution by learning from that feature. So each feature is parameterized with this probability, with this mean and standard deviation. So that's the concept. So
And sir, you have mentioned some operations are computationally expensive. So does it mean they take more time, or it is simply monetary terms they are expensive?
So when I use computational expensive, I talked about the molecular dynamics simulation or various simulation method, whether quantum or classical, because usually even with the advantages of GPU or CPU, you can do only few hundred nanosecond with a very good supercomputer with 60 or 70 GPU can only achieve 100 nanosecond in 1 day or 2 days. So that is what I mean by computationally expensive.
Okay. Sir, at one point of time, you have mentioned that for training of the model for small molecules, we have to go for RNA, smiles for converting into the machine readable pattern. In case of proteins or jumbo molecules, you have mentioned that we can go for graph VAE, that is autoencoder through variational. So is it necessary that for proteins we should go for only graph VAE? Or one more question, while training the model, do we consider entire protein structure, or only the active site we should consider for training the model for converting the structure?
We only consider the active site.
Okay.
So we used a graph-based approach here. I don't know. There is another paper which tried to drug design on the sequence-based. Just based on the sequence. So there is only another paper which is there, and I think only 2 or 3 paper. I don't know how they have done it, whether they have taken a RNN-based approach or a graph-based approach. So we took the graph-based approach to differentiate between the residues, because we thought that it will represent the active site in a proper way. So that's the reason.
Sir, one last question from my side. How different methods, that is molecular representation methods, how they decide the speed of the model training? Do they affect the model training? And what are the latest way to represent molecule in terms of more output in shorter period of time?
So you mean that whether it's a graph-based approach or a smile-based approach, or
smile, vector,
look, in speed, I don't think that it's a challenge for any of this method. But in recent phase, people has shown that the graph-based approach shows some improvement when you are calculating various toxicity profiles, or some of those properties, or various late-stage properties. So we are also trying to see the benchmark. So we are also trying to approach this problem with various methods to create a benchmark for ourselves, but I don't have a clear answer for that. But regarding the question of fast, whether it's a fast or slow, I don't think it's a big deal.
Okay, so because 2 papers are creating headlines in representation techniques. So one is by Jim, that is junctional trees, I think.
Exactly.
And one is by some MIT professor, that is message
MCTS, yeah, right.
MPN, message passing neural network, I think. And sir, you have mentioned that minimum 2 weeks are required to train a model as per the question of Dr. Prajwal. So 2 weeks means it is day night. I mean 24/7 you keep system on, or when you resume the office you again start from the last position.
I don't think I 2 weeks when I said it's. I said I took a bigger time. I mean it can be done within less than that time. Also,
Right.
you just run your system. I mean it's not a problem.
So you abort the.
But once you have your pretend model ready.
Okay. So, sir, there is one more concept I found in your paper, that is just to fine tune the last hidden layers while transfer technique is there. So, what is the logic behind tweaking those only last hidden layers?
Okay. So for the transfer learning, so we are only training our model with a big data set. So like CHEMBL or Zinc or something like that, so that it learn the grammar. So now the target specific data set is, we could also train our model with the target specific data alone, but we are not doing that because that data is not enough. Our model would not learn the diversity. So what is there, we are learning from the big data set. Right. So at the last layer, we are putting the transfer learning data set so that at the same time it learn those features as well.
Thank you, sir. Thank you very much, sir. Badekar, sir.
Yes, sir. Thank you, Dr. Arjit, sir, for delivering such an intellectual talk on accelerating the Novo drug design using deep learning approach on exploration of various chemical spaces, predictive specification, data digitalization, text mining for understanding various diseases and to improve health. Also, we have given so much of insights on AI, ML, DL and RL methods, future attribution methods, receptor binding methods in drug design using DL, so on and so forth. I'm sure all the participants has content with the deliberation of the session. We now proudly present certificate of appreciation to you, sir.
Thank you.
Thank you, sir.
Thank you. Thank you once again. Thank you. You are on mute.
I request Pradeep Bhavne sir to kindly present concluding remarks of this session.
Thank you, Wadekar sir. Today we had two awesome sessions. First was by Dr. Prajwal Nanikar, Senior Scientist, Schrodinger, on topic transforming drug discovery with advanced computational modeling. Second session was by Dr. Arjit Roy, Senior Scientist, TCS, Hyderabad, on topic accelerating de novo drug design using deep learning. These both sessions will help us to move ahead in this area of research. It will also motivate and inspire the young budding researcher to be the part of drug discovery using computational tools. The field of research without which it will be impossible to design a drug in future. We have witnessed how artificial intelligence actively involved not only in a drug discovery but also to solve the problems related to safety and ADME of the drug. As per my point of view, there are two takeaways from the session we had. First, artificial intelligence, machine learning and deep learning are the learning need to be added and inculcated in present to secure our future. Second learning is that before anything comes to the reality, it will be experienced using augmented reality, virtual reality and extended reality. It's true as per my part. Thank you very much, and with the due permission of the chair, I announce this session is.
Wait, please wait. Whatever, sir, are we taking the screenshot?
Yes, sir. I request all the delegates to please kindly switch on the camera of your device so that we can take a sweet, memorable picture for today's session.
With the resource person, right.
Yeah, yes, with resource person. Just honored.
I have made enable camera as well as the mic for all. Please switch on your cameras.
Yes, I'm putting all of it in a larger gallery
Switch on the
mode.
camera.
Can we accommodate more images, sir? Nanikar sir, if we
Yes,
remove,
I'm putting it on larger gallery mode right now.
if we hide the certificate, time being, I think.
Yeah.
Yeah, that's good.
We all are on larger gallery mode.
Yes, sir.
All are ready? One more, please. Thank you.
Thank you everyone for joining.
Thank you very much.
I request all the delegates to join tomorrow, okay, for the last session of this webinar at dot 10:30. We are going to proceed at 10:45 with Prajwal Nanikar.
Thank you, Roy sir. Thank you once again.
Thank you very much. Thank you. Bye.
Thank you, sir.
We are very sorry for a few questions we could not able to take due to the time constraint, but definitely the participants will get opportunity to ask question tomorrow. Okay, so if you have any question, you just email the question or put it on the Telegram group. We will forward your question and we will get back to you. Thank you very much. Have a nice day.
ती बघा की दिसती की पुढं कुणाच आवाज